welcome. I hope everybody who's on can hear me. I'm Dennis Hanasak. I'm here at Harbor Branch, and I want to welcome everybody to Indian River Lagoon Symposium 2021. Every year we have this, I give a brief kind of background and a welcome. And so I will do that as usual. Good. Hold on, just got to do a refresh. Here it is. So I always like to tell the story how this started. It started in 2012, and what triggered it was we had a meeting at Harbor Branch. We had a new president of FAU, and she was going around to all the campuses, and a bunch of us at each of the campuses would get up and talk about some research that was going on. And I was tasked to talk about Indian River Lagoon research, and I did. And at the end of the meeting, a guy by the name of Rick Minton, who was the head of the Harbor Branch Oceanographic Institute Foundation, came up and he goes, I really like that idea you had. And of course, I wasn't sure which idea right away. And I really quickly realized that what he was talking about was, I said, wouldn't it be cool if we could get everybody together, once a year and just talk about all the great research that was going on up and down the lagoon? And he goes, well, let's make that happen. And that led to dialogue with our foundation, who've always been very supportive, and they are our major sponsor every year, now for 10 years. And we talked to some other folks, colleagues at other institutions. We formed a first steering committee, and we had our initial meeting in 2012. Did we really believe that we could get as many people coming every year? Did we really think that we would have enough to talk about in year two? That was actually things that people were really worried about on the steering committee. We had this idea that maybe we wouldn't have enough ideas to have a different theme every year. But every year we have a theme, and our first one was the Indian River Lagoon looking forward, because we really wanted to take a really perspective in like what was going to happen in the next few years. And if you recall, that was just the beginning of what now may have been a tipping point in the lagoon. The super blooms had started the year before, and 2012, when that meeting was held, was actually before brown tides had started by a couple months. So we were right at the beginning of what many of us now recall as maybe possibly, like I said, a tipping point. And so, every year we meet here at Harbor Branch in the auditorium. That's from our first year. I see John Reed in the front seats. He was just here earlier watching from his office. And our tradition of having oral presentations and poster presentations goes back to that first meeting. So here we are. It's our 10th year. So, every year I like to look back. I'm not going to go through every year, but you can see that through time we have come up with different topics. There are some recurring ideas. If you look through there, restoration is one. Unfortunately, some of the challenges with those HABs, the harmful algal blooms, is there. Biodiversity is there. So it's a real mix. And you can see that we've had a range of keynote speakers. So we always pick a keynote speaker to match the theme. And then we kick everything off, in that fashion. And in 2014, we started in a very simple way to start to recognize students. And we thought, oh, well, let's come up with one award, and we'll do it, for the best posters. We'll encourage the students to get posters. And it was really the first time we did that. And the funny thing that happened in that year was we had a bunch of judges, and they were all making their decisions, and a couple of them came up to me and they go, we have way too many good posters. So literally, I got $200 donated from the people in the room so that we could give 3 posters that first year instead of 1. And if you look at the history of student achievement awards, the following year, we started oral presentations. And if you look through time, we started to increase the number of special awards. And then 2 years ago, in 2019, we started the People's Choice Awards, which was everybody gets a vote on the 3 different types of formats. So last year, and the plans for this year are, we're going to recognize 9 students for their excellent research. An important part of the symposium has always been to foster career development. And if you remember from all of the calls over the years, we do say that we favor presentations by students and early career professionals. We do always have a nice mixture though. And so here we are, year 10. So last year at the end of the meeting, I said, boy, I've got a great idea. Stay tuned for our 10th symposium. Well, along came the pandemic. And so our plans did change. Our plans changed to the point where the steering committee thought, maybe we shouldn't be doing this. It's really hard to do what we do every year in the symposium, get 350 people together, spend the whole day totally immersing ourselves with a lot of dialogue and a lot of socializing. And I said, yeah, I can't imagine doing it. It's just not going to work, and it's not going to be the same and all that. And so I was willing to defer this until this fall. I was even willing to defer it for a year if need be. But then, funny things have happened in the year of the COVID, and we've all learned to adapt. And really what happened is one of the members of the steering committee said, I was good with the decision, but one of my students came to me and she said, I was really looking forward to the symposium this year. I'm graduating in the spring. And if I don't present this year, I'm never going to present at the Indian River Lagoon Symposium. Well, some of you may know I actually have a soft spot for things like students. And so that made me start to think about it. And the steering committee started to revisit the idea. And then also, I realized, as did all of us, that actually Indian River Lagoon research continues during the pandemic. Some of us have still been doing field work, pretty much the way we always have. Lots of social distancing out in the lagoon. It's a big place, right? So that's not quite so challenging as working in the labs. A lot of our colleagues were kind of stuck at home, but many of them came up with really clever ways to continue to be productive. Sometimes they were working on data or publications or proposals or some combination of that. And in fact, there's been a number of new research initiatives that have come up in the past year. And then I think the last point was, as I was doing some work up in the northern IRL, seeing the latest version of the blooms up there. And of course, we all recognize how dynamic the lagoon is, how it varies from year to year. The lagoon didn't shut down during the pandemic. All the issues are still there. All the research needs are still there. And I think, to me, this was the most spectacular year for the lagoon. It started out that way. In March, it was the greatest visibility I've ever seen in my life in the lagoon, in local waters. I saw things I've never seen before. And then, as I mentioned, I saw the very continuing change of the blooms to the north. So, for all these years, the symposium has been a way to take a look every year to see where we are, to kind of take stock of the research, to go over old projects sometimes, to have new collaborations and talk about, what students will be doing. And then the following year, we get to see what they did, for their projects. So anyway, we decided, let's do this. And once we decided to do it, we decided, let's try to go all in on this as much as we can. So just the other day, as I was driving to work, I realized, the last time I had been to a scientific meeting in person was in fact the symposium. And then I started to ask colleagues, and I started to realize very quickly that for most of us in this virtual room today, the last time we were all together at a meeting was probably the symposium. So in the end, while our auditorium today is quite dark, I think that we're going to try to shine a light on Indian River Lagoon research today. So this is one of the reasons why we encourage you to turn on your cameras, not off. You can multitask some other day. We do want you to be muted because muting is necessary because there's so much feedback that happens when somebody has an open mic. We will try to patrol that, but it's really hard with so many people. So please always mute unless you are the speaker. And lastly, you know, most of these meetings that are much smaller than this, they shut the chat box off. You don't have any idea who's in the room. You should be able to see everybody in the room who's there. You should see the participant list. You have free access to the chat box. That is how we will ask questions for the presentations, but also you can use it for anything else you'd want to. You're more than welcome to use private chats. If you see a colleague across the way or somebody you don't know and you want to say, hey, go for it. So we really want to be this as interactive as possible. So this year's theme addresses IRL research that is being conducted during, and I would argue, in spite of the COVID pandemic. Short overview of how we're doing this. If you've been here before, it's the same format. We're going to do exactly what we've done before. It's just that we're using the virtual space that, you know, most of us have done that a lot. So we're going to have our keynote speech as soon as I finish my remarks. And then we are going to have an oral session. And the way we have done this, and actually if Bob Chamberlain's out there, he was the one on the steering committee many years ago. He said, yeah, first oral session, we ought to try to get it as close to the theme as possible right after the keynote. So Bob, if you're out there, that's something you first suggested at a meeting and we still do it. Then we're going to take a quick break. Virtual breaks are usually quicker than meetings when we're all gathered around and we're all trying to get into the same restrooms and the same pot of coffee. But hopefully you all have your own accommodations already reserved. We're going to have a session on IR restoration, which, as I said, is a recurring theme. We're going to hit those 3-minute, 3 slides. That was something in the first year we started, people were very reluctant to do. I actually advocated for that, even though I had thought in the past it never worked right at other meetings I'd been out. But as I think you all know, it works out really well. And so now the last couple of years, including this year, we have 3 sessions. So we have about 27 of those talks. And then we're going to do poster sessions, which you know, you've got emails about, but we'll talk about that later. And then tomorrow, it's going to have the same posters presented a second time. The reason for that is so that everybody will have an opportunity to see more posters. The posters presenters will have an opportunity to get more comments and discussions going. The judges who are judging the students will have a little more time and flexibility to do their evaluations. So then tomorrow, we also have 2 more oral presentations. Again, common themes, water quality, human impacts, IRL biodiversity, 2 more short sessions. And then the other thing that we decided to do this year is every year we give out the awards, you know, at the end of a very long day and after a reception. And some of the folks drive, you know, a couple hours to get here. And sometimes they're driving and, you know, students are coming together with their faculty in a bunch of vans. They got to be back. And so we always have a lot of people at the student awards, but not as many as we should. So our hope is that we can really truly recognize our students easily in a virtual platform. So we shall. And if you need something else to encourage you to come to that, for the first ever time, but because it's our 10th anniversary, we shall be doing the IRL Trivia Challenge. So yes, we're gonna have some fun. If you know anything about my mantra here at Harbor Branch is yes, we do our research hard, but it's okay to have some fun. So that's meant to be total fun time, and we will have some awards for that as well. So we hope everybody will not forget about Friday afternoon. You know, it's impossible to do this without a number of really great people. Every year the steering committee changes a little bit. I put everyone's name up again. Normally I say, please stand. So I guess they could stand right now, but it wouldn't be the same as standing up in the auditorium. But I want you to note those names very carefully. They're on your program. They're listed in a lot of different places. So if you have any comments, suggestions, let them know as well. If you know somebody well in there, you'd rather talk to them, go for it. You can tell any of us what you think. We always do a little debriefing after each symposium and we start to plan for the next one. Okay, but I just want to say that they do so much work. For example, a subcommittee today and tomorrow is organizing all the judges. They take care of all that. And just about everybody on that list will be moderating a session. And during the whole planning and post analysis time, I reach out sometimes to all of them, or just individuals who I know a topic really well, and I get their input. So it's really great. We do have great sponsors, and 2021 is no exception. As I mentioned earlier, I told the story of how this got started, and the foundation has been really good. And they have a fundraising event every year, and they are doing that also this year for the first time virtually, the Love Your Lagoon event. And you will see all of the names of the major donors to that event. And that has always been used to support this event, and also to support graduate student research. I do want to say the highest level sponsors, and I think they've been this for probably every single year, are the Dukes, Joe and Jennifer, and the O'Reillys, Michael and Marie, and Marjorie Raines and Harold Baker. And again, please read the program. Everybody is recognized. And also, this is, I think, our third year where we've been supported also by the Indian River Lagoon National Estuary Program. And I should say that also they are supporting the proceedings from last year, which, remember, we had a very special biodiversity meeting. And I will send out announcements on that, but those proceedings will be coming out as planned in May. And with that, I am completed. And I am going to next go right into introducing our keynote speaker. And I always write up, these introductions, and of course, I don't have that with me right now. So I'm going to have to totally ad-lib the introduction to Dr. Ed Flips. Ed went to school. He got his a bachelor's degree in the early 1970s at the University of California, San Diego. And he went to school for his master's and his PhD at the University of Miami, and then did a 2-year postdoc. That's actually when I first met him on a visit down there. And shortly after that, 1983, is when he moved to the University of Florida, and he went through the ranks and is now a professor. So he's had a long career at one institution. I know what that's like. It's pretty good if it works, and it's worked really well for Ed. Over the years, Ed has his work has gravitated more and more toward looking at phytoplankton in Florida waters. He's worked in St. John's River. He's worked in Florida Bay, the Swanee River estuary, and of course, and not the least, in the Indian River Lagoon. And in 1997, he started out what has now become 23 years of phytoplankton monitoring in the lagoon. And I think when Ed first started that, I think a lot of people thought, oh, it's algae, it's phytoplankton. Not a lot of interest at the time. And I think that, what really drove Ed, that drives so many of us, is curiosity. And his curiosity, was in that group of organisms. But now, 23 years later, he has quite a story because of what he started then. And I think he'll tell us a little bit about how that started. And he's also going to tell us a little bit about what he's doing now. So I think he will give us a wonderful keynote address. And I will say that what we decided to do was to go ahead, have him tape it to make sure that everything came across exactly as we would, so there'd be no connectivity problems from Gainesville. But he is here at the meeting, he is live, and he will take questions at the end of his talk. And that is the only one that we've taped, but he is special, he's the keynote. Ed's talk today is going to be a 23-year view of algal blooms in the northern Indian River Lagoon, cyclical patterns, and a regime shift.
Hello, and I'd like to first start off by thanking the organizers of the symposium for giving me the opportunity to talk to you about our 23-year so far journey into phytoplankton blooms in the northern Indian River Lagoon. There's a lot of people who worked with us on this project, and I've just listed a few names, but there are many more, and some of the major people who have worked with us and for us on this project. One of the big organizations, of course, St. John's River Water Management District, who were funding the project from the very beginning in 1997, are continuing to help us out now. The Indian River Lagoon NEP, National Estuarine Program, for their funding assistance and involvement in the project. The Florida Wildlife Research Institute, who also provided funding for this in collaboration. And of course, University of Florida and IFAS, who provided funding for us. I'm going to divide this talk into two parts. To paraphrase Charles Dickens, part one will be A Tale of Two Lagoons, which will focus on really the first 10 years of the work we did. And then part two, Great Unexpected Surprise, which I'm going to tell you about because I would ruin the surprise. The culmination of the first 10 years of work that we did ended up in a meeting I was invited to go to on long time series observations in coastal ecosystems in Rovinj, Croatia, which is pictured here. And it turned out to be illuminating in terms of how different parts of the lagoon operate. And I'll go and look at that in more detail with you right now. This research that started in 1997 focused on 10 primary sites, and you can see them listed here, from Vero in the central Indian River Lagoon up to the Mosquito Lagoon, Oak Hill and Mosquito Lagoon. And the northern range that we're talking about in terms of the focus of a lot of phytoplankton blooms, it really extends from Melbourne northward into the southern Mosquito Lagoon, including the Banana River. Now, to make things a little simpler, I'm going to focus on two sites, one in Titusville in the northern Indian River Lagoon and one in Sebastian in the central lagoon, because otherwise the talk would be many hours in length. So let's focus on those two sites and see how they look the same or different. This figure shows a time series of phytoplankton biomass from 1997 through 2008. And let me just introduce you to the axes so you're familiar with what we're talking about. The y-axis is phytoplankton biomass as micrograms of carbon per mil. That unit is, you don't often see this unit, but it's roughly equivalent to 1 microgram carbon per mil, approximately 10 to 15 micrograms of chlorophyll A per liter, if you want to get some perspective. So that means that along this time series, these large peaks that you see from 2002 to 2006 actually represent chlorophyll levels in the 60 to 70 micrograms per liter range. And that's very high when you consider the fact that most trophic state indices for coastal environments would indicate that anything over 20 micrograms per liter would be high and, you know, representative of eutrophic or even hypereutrophic environments. Another feature that you'll notice is that from years 2002 through 2006, the peaks were much higher than in previous years. And that also correlates to La Nina and El Nino conditions. El Nino conditions being years when you typically have higher rainfall, particularly in the winter and early spring. And so this is a trend that we'll see again later in the talk. The other thing you see is that the dominant bloom forming species in this particular case, and in many regions of the northern Indian River Lagoon, because this is similar and representative of most of the lagoon in those regions, was dominated by the Dinoflagellate Pyridinium bahamense. And you also see a diatom bloom, fairly significant diatom bloom. And we'll talk a little bit more about that. Now the figure also shows you the relative biovolume, or actual absolute biovolume of dinoflagellates, diatoms, cyanobacteria, and other groups, so you can get an idea of what kind of dominant structure the system has. So let's talk for a second about Pyridinium. Pyridinium is an important bloom former, probably the most important one over long periods of time in the northern Indian River Lagoon, but it's also a really important species in other parts of South Florida. It's a true tropical species, and it's also a saxitoxin producer. And therefore it poses a risk for the presence of toxins in fish and invertebrate species where blooms occur. And that's why the FWRI actually classifies Paradinium as one of the two species they monitor very closely in terms of the presence and potential exposure to saxitoxin, which is a neurotoxin. In Southeast Asia, Paradinium is really important also, and it can be the cause of numerous human fatalities every year because of the consumption of unregulated shellfish. Let's take a look at the other lagoon we're looking at. That is the Central Lagoon, in this case Sebastian, the site in Sebastian. And you can see that there's none of those major blooms that we found in Titusville. In fact, there were never any observations above 1 microgram of carbon per mil. So obviously this system is operating quite differently, even though it's connected to the northern Indian River Lagoon. So why is that, do you think? So let's take a look at what's similar and what's different in these two areas of the lagoon. Well, of course, the temperatures are very similar. They're not that far apart. Both are similar range in temperatures. A little bit cold, a tiny bit colder in the very northern parts of the lagoon, but not very different. Light availability, which is often dictated by mixing depth, is similar. Both regions are typically 2 meters in average depth or mean depth, and so there's really no difference there. But where there is a big difference is in water residence time. And this shows the E50, which is the time it takes to replace half of the water in a basin. In the northern Indian River Lagoon, it takes over 2 months, and that's true of the southern Mosquito, Banana, and northern Indian River Lagoon proper, and sometimes well over 3 months. So these are very long water residence times. In the Central Lagoon, residence time on the order of days, certainly less than 2 weeks. So much shorter, much less time for blooms to develop. But the additional factor is also Atlantic tidal flushing, which actually dictates some of this residence time issues. But in the northern Indian River Lagoon, there's obviously very low levels of flushing rates. But in the Central, there's high, and that makes a difference, and I'll show you that in just a second. The nutrient differences are reflected here in this particular table. And these are mean nutrient concentrations over that first 10-year period. In the Sebastian area, TPs are actually higher, average TPs are actually higher than in Titusville or Banana River. Even though the differences aren't large, it does reflect the fact that the Sebastian area does receive significant watershed inputs from agricultural areas and other areas, so their loading rates are quite high. And that's reflected, for example, in the Sebastian River inputs. If you go northern, the watersheds are smaller, although they're still nutrient rich. If you look at nitrogen, actually the nitrogen levels are lower in Sebastian, average nitrogen levels, about half or less than what they are in the northern Indian River Lagoon and Banana River. And part of that has to do with the fact that the water coming in from the Atlantic is very low in nitrogen. You can see that the Cape Canaveral shelf environment just offshore, which these are values that we came up with in a 6-year project in the Canaveral region, the concentrations of nitrogen are quite low. And TP levels are lower also than the average levels in the lagoon itself. So you can see that Atlantic flushing has an influence on the nitrogen levels significantly. The difference in the nitrogen levels from the central to the northern lagoon are reflected in work we did with nutrient limitation bioassays of phytoplankton growth, which indicated that in the Sebastian Central region, nitrogen limitation was a predominant limiting nutrient when things were limiting, but in the northern part of the lagoon, there was more evidence of phosphorus limitation or co-limitation with nitrogen. Now for the supplies, which you've all, I'm sure, been waiting for. I think the one thing before I show you the results is the fact that we tend to think that 10 years is a long time series, and we understand a lot about a system, and we certainly did learn a lot the first 10 years. But ecosystems are complex and can be subject to rather dramatic changes which were unexpected, and that's certainly what we had in this case. So here's the image of the full time series from 1997 to 2020. And you can see the line of demarcation is 2011. And you've already seen the first part, but all of a sudden we started to have blooms of 3 or 4 to 5 times higher magnitude and more prolonged and prolific in terms of time frame as well. In addition, they were contained more often than not different species, small flagellates, other small species of nanoplankton, including the brown-tide species Areumbra, which was the one pictured in the last frame, cyanobacteria, other nanoplankton, and the Paradinium still was present, but you can see that these other species became more dominant. And this is representative of not just Titusville, but the other major regions within the northern Indian River Lagoon, including Banana River and the southern Mosquito Lagoon. You can see by the scale on the left that blooms got up to 15 micrograms carbon per mil. And in other regions, they were actually up to 20, but the pattern was similar. So this was quite shocking because the chlorophyll levels in these really big blooms reached 200 micrograms per liter, hypereutrophic by any definition. So let's talk about what happened, what may have caused it. First of all, I'd like to show that the El Niño La Niña pattern that we saw in the first 10 years continued. Until 2011, the El Niño periods were higher in average biomass than the La Niña periods. The same occurred after 2011, but at much higher average biomass levels. So why were they that much higher? So the first place we typically look would be nutrients. There must be something supporting these very high levels. And indeed, if you look at the time series of total nitrogen and total phosphorus, you do see a big change in 2011, particularly in phosphorus. The baseline of total phosphorus went way up significantly, and the peaks also went way up. And that again coincides with what we were seeing in the phytoplankton. And so where did this extra nitrogen phosphorus come from? But particularly the phosphorus. And let's take a little more of a deeper dive into that. This figure shows the annual average TP versus annual average phytoplankton biomass at the 3 regions within the lagoon, 1 being the southern Mosquito Lagoon, 2 being the northern Indian River Lagoon, and 3 being the Banana River. But the trends show 2 important things. One is that the total phosphorus levels were higher, the range was higher in the 2011 to 2018 period. We're adding additional data for a paper we're doing, but for this iteration, we go through 2018. And also the slope of the curve is much sharper, indicating that the phosphorus levels were higher, but also the conversion to biomass also was quite efficient, which indicates with the significance of the relationship that it appears like the added phosphorus was one of the main driving factors for this enhanced biomass of phytoplankton. If you look at this particular figure for TKN, and average phytoplankton biomass, similar trend, except this is a little different. The actual upper range of TN values are not really significantly different, even though the lower range is a little bit less, 2011 to 2018. And the slope, again, is increasing, but you can see that the significance of the relationship is slightly weaker, which again indicates that phosphorus has become an important player, even though nitrogen may also play a role. So let's start with the transition from 2010 to 2011, because that was the first time we saw this extended and more profound increase in the bloom. And this figure shows something interesting in terms of what part of the driving force for this might have been, and that was the demise of the benthic primary producer biomass from 2010 to 2011. And this was in a paper from 2016, and the data was provided to me by Laurie Morris and Robert Chamberlain from the South Florida St. Johns River Water Management District. But you can see the dramatic decrease in primary producer, benthic primary producer biomass from 2010 to 2011. And interesting, when we did a rough back of the envelope calculation of the change in dry weight of these producers, primary producers in the benthos, that it kind of correlated quite well with the amount of increased biomass in the water column of phytoplankton, indicating that there might have been a correlation there. So this may have been one of the initial drivers of that bloom phenomena in 2011, was the greater availability of nutrients being recycled from the dying and diminishing populations of seagrasses and drift algae. So what caused this demise? And of course, this is speculative. There are a number of different theories about this, but part of the reason may have been the exceptionally cold winters of 2009-10 and 2010-11. And you can see that in this figure. And during that time, during those really, really dramatic cold snaps, there were observations of mass mortalities of fish, of invertebrates, and that may have contributed to the stress on these benthic primary producer communities, which allowed the phytoplankton blooms to occur through the added biomass degradation and entry of available nutrients, nitrogen, phosphorus, into the water column. Another observation was that this period of time around the demise of the seagrass populations was also a period of high salinity in most of the region. The high salinity was associated with a prolonged La Nina period, and the question is, do salinities at this level really stress the seagrasses sufficiently? Well, it's possible with multiple stress events, including the very low temperatures and the salinity together may have contributed to it. But the other thing it shows is that the high nutrient concentrations observed in 2011 were not because of a period of really high external loading of nutrients, which typically are associated with wet periods or high rainfall periods. The other important change that happened in 2011 is that after 2011, these big blooms, very intense blooms, which prolonged in intensity in terms of time frame, also resulted in very low light transparency through the water column. And I show this figure as a Secchi this depth, and you can see the line at 0.5 meter Secchi depth. That would be roughly equivalent, the Secchi depth is roughly equivalent to the depth that's required for seagrass to survive. And so you can see that at 0.5 or below 0.5, seagrasses are going to have a tough time doing well at depths greater than 0.5 meters. And you can see that the Secchi's depths in some of these periods were as low as 0.1 meters. So that means that not only were the blooms, there for the first year, but the subsequent blooms and returning blooms every few years probably stressed the seagrasses and prevented them from coming back or reestablishing themselves because of very limited light conditions. So overall, let's take a look at an oversimplification, as schematics generally are. But I think this schematic provides you a very rough idea of what I'm just talking about. Before 2011, external nutrient loads were obviously major drivers of production for phytoplankton, drift algae, and epiphytes on seagrass, and even seagrasses to some extent themselves. And there was also, of course, internal load coming up from the sediments. And that also was probably driving certainly seagrass epiphytes and to some degree the water column nutrient levels. So with those two loads, external and internal, you are driving phytoplankton, drift algae, seagrass epiphyte communities. You have a balance. In some years, probably there's higher phytoplankton. They're taking advantage of the external and internal loads more efficiently for a variety of reasons, and you have more intense blooms during the low rainfall years. You have lower external load and less availability of nutrients for phytoplankton. In the post-2010 period, you have a change. The seagrass communities are gone. They really haven't come back even to this date. And so you're missing the seagrass and epiphytes, which are not only absorbing the nutrients, but also competing for the epiphytes, competing for the nutrients in the water column. They're gone. Phytoplankton blooms are very intense. Therefore, there's not much light getting to the bottom, therefore preventing the reestablishment of these benthic primary producer communities, therefore somewhat less drift algae biomass as well, but greater internal load, because once you lose this stabilization of the sediments by the seagrasses, you're increasing the opportunity for nutrients in the sediments to come up into the water column. And the external loads probably have not changed all that much in terms of their influence of phytoplankton, but now all the nutrients from that are going into phytoplankton. And that's probably an oversimplification of what's going on. There are obviously many other factors, top-down control of blooms and how that influences the phytoplankton dynamics. There are other factors to be considered, but this is kind of a rough description of how these relationships may be driven in terms of the very large blooms we are seeing after 2010. Finally, to just reinforce what I've been talking about a bit, I've shrunk the time series for phytoplankton biomass for the Titusville site. And you can see a couple of things going on here. 2015-16 was a period of high rainfall. It also was a period when we had two major hurricanes influencing the region late in the fall, in the early fall, I should say, of 2015. And you can see that was followed by major bloom period extending all the way from the late fall of 2015 into the winter of 2017. And that also illustrates one of the problems that blooms create, because seagrasses, drift algae, very often their maximum periods of initial growth and expansion are in the spring, early spring through late spring. And of course, if you have blooms that extend all the way from the winter through the summer season, this really prevents those groups from taking advantage. The one hope we see from this figure is the fact in 2017, which was a low rainfall year, that there really were no major blooms in this particular region. And so there is some hope, but there is also a warning that what we're really seeing is a regime shift. And reversing it may be very difficult. And that's kind of the final message. So we are dealing with a regime shift in the structure and function of the northern Indian River Lagoon, including the southern Mosquito Lagoon and the Banana River. Actually, the central lagoon in terms of phytoplankton dynamics have not changed dramatically. And that tells you, of course, an important message. Can it be reversed? Of course, that's for another talk, but there are a range of different research projects going on as we speak, funded in part by Indian River Lagoon National Estrogen Program and other organizations to come up with solutions. It's a challenging task because in an area where there's rapid population growth, and of course we're dealing with global warming, which increases the potential for blooms in the winter, that it's going to be a difficult task that we face. Thank you. If you wish, I'll be on live to answer any questions you may have.
Okay, so if you have any questions, we have time for a couple. If you can type them into the chat. I'll see if I can find them.
I'm here.
Hi, Ed, are you there? Can we see you?
I'm
There you
here.
are.
Can you hear me?
Yes, a little louder maybe on your end.
Oh, okay.
So this is from Maria, and she asked, is the substitution of the dinoflagellate species by the Ariorumbra a shift that has happened in other parts of the world?
That's difficult to say. I'm not sure if there's a great deal of data available on that shift that I've noticed. I think the distribution of Paradinium and reports of Paradinium and the brown tide have been coming in over the last few years from different parts of the world, but I haven't seen a report of a linkage between that shift. And one thing that I think it's important to keep in mind is that actually the Paradinium hasn't gone away. There are parts of the lagoon where there have been some really dramatic Paradinium blooms. They tend to be shorter in duration than some of these brown tide events, but they're still there. So they're responding as well, but they're a true tropical species. So typically they would be blooming in the warmer months of the year, so that if an Arioumba or another nanoplankton takes off earlier in the season and start to sequester all the nutrients, then it's difficult for Pyro to compete. But there have been big blooms of Pyro too.
Okay, here's a question from Malcolm McFarland. He's also a phytoplankton biologist, for those of you who don't know him, but I know Ed knows him. What is your opinion on efforts to enhance flushing and reduce water residence in the northern IRL?
Yeah, and there are people working on that. I know FIT and I think FAU, some of the people at FAU are working on that. I mean, I think one of the two ways you can look at controlling the blooms, obviously, is one is to control the amount of nutrient availability for the organisms that are blooming, and the other side is to increase loss processes. In other words, increase top-down pressure, which is difficult to do, called biological control, or reduce water residence time significantly enough in order to prevent accumulation of biomass. And I guess the idea is to bring in water from the Atlantic, which is low in nutrients and low in phytoplankton, and hopefully flush out the system at a rapider rate. So I'm not a hydrologist, and I know that people are working on the feasibility of this. I think it's an important study, and it's certainly in a very, very broad thematic way. I think it's an appealing concept. And so hopefully they'll be able to model this and test it out and see if it's actually going to work without any serious collateral damage, which is true of any mitigation effort.
The next question is from Nikki Dix, and you probably know Nikki. She's up at the GTM, but she also got her degree at UF, so you very likely know her.
Yeah,
Can you discuss the role of organic versus inorganic nutrients in plankton community changes?
Yeah, well, and that, of course, that's a little more detailed question, but yeah, it's important in this case, particularly because there are species like Arioumba, which do a very efficient job of utilizing organic nutrient compounds. And that's true of a lot of different species of nanoplankton. A lot of nanoplankton are actually mixotrophic. And so as a consequence, when you shift over and you have high levels of organic compounds, that would enhance the competitive ability of things like a brown tide species. And of course, people have proposed that and studied that in different parts of the world, including like Laguna Madre in Texas, and have looked at that to some degree. And so, yes, it's an important issue.
For the applause.
And it's something
that
will drive the competitive relationships between species, the relative balance of inorganic and organic nutrients.
Okay, maybe one more from Chip. Do you think that the massive increase in filter feeders like oysters and other shellfish will be an important part of the solution?
Yeah. I mean, it's kind of the same question, of course, as in the case of flushing. Of course, is it feasible? One, is it feasible to increase the population sufficiently to actually have an impact on the algal blooms? And I know that there's people working on oyster restoration and clam restoration efforts to increase populations. And certainly filter feeders can process a lot of biomass and are potentially active in terms of top-down control of blooms in certain areas of the world. Whether we can increase the population sufficiently in these areas to have an impact, I think is a question for someone else. It's intriguing, and that would be nice if we can increase. I think one of the questions, of course, is there may take more than one approach to deal with this issue, and that is taking multiple actions in different directions to cumulatively have a significant impact on bloom dynamics. And so bringing nutrient levels that are coming in is important. Flushing may be a contributing factor, especially for certain regions where you can do that. Increases in top-down through enhancement of filter feeding bivalves could have an impact. So hopefully together, if you can bring these together in the right combination, it might actually help significantly.
Thanks, Ed. I really want to applaud you. I mean, I guess more figuratively, because we were all in the room together. You'd hear a resounding applause from us all right now. I want to encourage you to, in the audience, to continue to send some questions to Ed. I see another one just popped up. So Ed, if you're willing, you can continue to answer some questions in the chat. You can send them to all if you wish, or you could just respond to the questionnaire. That's up to you all. During the meeting, we really want to encourage that dialogue so it can continue after a particular presentation has passed. Thanks again, Ed. I hope that in 23 years, we'll have a 46-year record that you can come.
Okay.
Or you probably want to be virtual then, but you can come and do that talk, and we'll look forward to that too.
Well, let's say 25 years, in a quarter century.
Okay, we'll round it up.
but yeah, thanks. And if anybody wants to ask me a question, just send to my email, flips@ufl.edu. Not Phillips, though. That's the provost, and he doesn't know anything about algae. So make sure it's Flips. Thanks.
Okay, thanks again, Ed.
Thanks.
And now we're going to just say goodbye to Jeff Beal, who is the moderator for the first contributed oral paper session. And remember, these are 10 minutes, and I will just say on behalf of everybody that the speakers have been well coached. A lot of them have practiced earlier with how we're doing this. They know they have a 10-minute slot. That's it. We've told them go for 8 minutes, and you'll be done and totally done with questions by 10. Okay, Jeff, you're up.
Thanks, Dennis. Afternoon, everybody. Hope you're all safe and healthy out there. It's good to be together in a sense. First up, we have a wide cross-section of disciplines in this session entitled A Taste of IRL Research During the Touch of irony in the title from Dennis. The symptom of the virus is in fact the loss of taste for scientific discovery, and that'll be on full display in this session. So first up, we have Stephanie B, and she's going to tell us about novel cyanobacteria.
Thank you, Jeff. Today, I will present the results from the FWRI team about the cyanobacterium that bloomed in the IRL during the summer and fall of 2020. Let's start with an overview of the sequence of bloom events. In August of 2020, we started to receive widespread reports of green water discoloration. About the same time, we started noticing unfamiliar cells at our regular monitoring sites. By early September, the alarm was sounded for collaborative efforts to intensify monitoring. Reports of low chlorophyll were not compatible with observations from the field and lab. The culprit was determined to be an unidentified cyanobacterium. At this point, extensive monitoring proceeded with efforts to perform cell counts with light microscopy, cell counts with flow cytometry, toxin analysis in cooperation with Florida Department of Environmental Protection, and the determination of the molecular signature of the unknown organism. Identification efforts are ongoing despite abrupt decline of the bloom by the end of December. These satellite images from Sentinel-2 illustrate the non-bloom conditions back in the spring on the left, the striking water discoloration and spatial coverage of the bloom in September in the center, and the system at the end of December approaching the bloom decline on the right. Between September and January, 264 samples were analyzed using light microscopy collected from 60 sites at a depth of 0.2 to 1 meter. The smallest circles indicate sites visited only once. The larger circles indicate sites visited 21 to 23 times during this period. From these 60 sites, 13 sites were most frequently sampled, at least 5 times. Of these 13, 7 most frequented sites were chosen for comparison with flow cytometry data from September 10th and 15th. Of these 7, 3 sites were analyzed for toxins by FDEP for select dates. Light microscopy examination revealed an unfamiliar cyanobacterium. On the left, you see a light microscopy field of view of a live sample on top and a Lugol's preserved image on the bottom. To the right are details of the same images, providing you with a sense of the challenge we faced to count these samples and the striking difference in appearance of cells upon preservation. Cells observed in live water samples were round to oblong in shape, approximately 3 to 4 micron by 5 microns, with a pale translucent green appearance. Cells were most often seen dividing into chains of 2 and up to 5 cells in length. The most striking feature was the presence of elongate aerotopes, seen here as this squiggly structure, which are gas vacuoles typically characteristic of cyanobacteria. Upon preservation with Lugol's solution, the cell changes in shape, becoming more capsule-like, and the content becomes more homogeneous. These characteristics point to cyanobium and cyanobacterium as likely candidates, but it is possible that it is currently undescribed. Examination of the thylakoid arrangement using TEM and molecular markers would be needed to differentiate between these 2 possible genera. The fact that chlorophyll levels in the field were very low to not detected was explained when flow cytometry revealed the dominance of cells with a high phycocyanin-like signal. This result was visually confirmed by epifluorescence microscopy that showed a lack of emission under chlorophyll wavelength excitation. However, examination under phycocyanin wavelength excitation revealed the dominance of cells with red emission. Based on data from 7 sites analyzed for 2 time points, it was also important to demonstrate early in our extensive monitoring that light microscopy and flow cytometry counts were within the same order of magnitude. We found the Pearson correlation to be statistically highly significant. Light microscopy counts were performed using the settling technique and the inverted microscope. The maximum concentration found reached 3 million cells per milliliter. We started to see consistently elevated cell concentrations of the unknown cyanobacterium in early September. This prompted us to go back and recount archived samples from the area to see if this organism was present before the bloom occurred. The earliest detection of in archived samples thus far was June 23rd. We plan to go back even further using cell counts and flow cytometry to investigate when it may have first appeared. By mid-December to early January, we start to see a decline in cell concentrations. Samples from 3 sites taken at 5 time points were analyzed for toxins by FDEP and returned non-detected to very low concentrations for all targeted toxins. We looked at spatial distribution over a period of 20 weeks between September and December. Bloom concentrations started appearing initially in the Indian and Banana rivers in early September, with 5 sites having concentrations over 1 million cells per milliliter, represented by the largest circle. 2 weeks later, we started to see it popping up at more locations, including sites in the Mosquito Lagoon. In week 5, it is detected at our southernmost site in the Indian River, with the highest concentrations persisting in the Indian and Banana River basins. Between the weeks of October 18th and November 15th, we see an increase in sampling efforts with up to 20 samples collected each week and our most expansive spatial coverage from the northern Mosquito Lagoon stretching south of Melbourne. In week 13, we finally start to see a decline in cell concentrations in the Indian River Basin, but it is still hanging on in the Banana River. Finally, during the last week of December, we see the concentrations drop to below 10,000 cells per milliliter in the basins where the bloom initiated. We currently do not have elements to pinpoint what triggered bloom initiation. As Dr. Flips pointed out in his talk, regime shifts may have allowed this cyanobacterium to take advantage of a possible open niche in this system when examined over a broader timescale. However, we can suggest a possible driver for bloom decline as we observe weather conditions during the bloom period. Cell concentrations and monthly average air temperature rapidly declined at the same time. The best fit curve for water temperature during this period shows a clear decline, and the Pearson correlation between cell concentration and water temperature is statistically significant. This signals to the fact that this species, as many other cyanobacteria do, has a strong affinity for warmer water conditions. Understanding the molecular makeup of this cyanobacterium started with direct PCR sequencing of field material targeting rRNA that led to a 400 base pair fragment. This material was further sequenced using universal 16S primers and newly designed primers that generated a 2000 base pair fragment. From there, we took 3 different approaches: qPCR analysis, amplicon sequencing, and metagenomic sequencing. With the qPCR, we have already inspected about 60 samples across the bloom period. This time series of samples will help us correlate sequence abundance with microscopic abundance. Amplicon sequencing has targeted the full 16S, and metagenome sequencing has focused on the whole community genomics of one sample. Information from the amplicon and the metagenome approaches are being used to calibrate and expand the qPCR capability. Once we become confident of the correspondence of sequence abundance with microscopy and flow data, qPCR will become a powerful tool to inspect archived samples to better understand possible distributional trends of this cyanobacterium prior to the 2020 bloom. Moving forward, qPCR can provide early warning of its presence at very low levels. Finally, the metagenome analysis provided a full 5.8 mega base pair genome that is in 100% agreement with our previous PCR sequencing. We are now in the progress of annotating the genome, which will help us to understand the physiology and phylogeny of this cyanobacterium and hopefully propose its identification. With that, we would like to thank our many partners and colleagues. And to learn more about the details of this bloom, be sure to attend the speed talk by Tim Moore later in the symposium. Thank you.
Thanks, Stephanie. We're going to go with just one question from Nikki. Stephanie, what were the chlorophyll levels, and would you recommend we start monitoring phycocyanin more frequently?
I do not know the actual chlorophyll levels. The low chlorophyll was something that was circulating from other agencies that was reported basically in the field and possibly in the lab, but we didn't have exact chlorophyll levels.
Very good. Thanks.
Yeah.
Next up, we have a student presentation, so judges, beware. We have Adam Searles. He's going to tell us about trophic competition in 2 fishes.
Everyone. Can you all see? All right. If we're good, I'm going to go ahead. My name is Adam Searles, and I'm a PhD. Oh, we all good?
Yes, having a little trouble hearing you, Adam.
Oh, okay.
Get a little closer.
Okay. Hi, everyone. My name is Adam Searles, and I'm a PhD student at the University of Florida in the School of Natural Resources and Environment. And my current research focuses on the dietary overlap between Western Atlantic sea bream and sheepshead in the northern Indian River Lagoon. So to set the context for my project, Climate change is facilitating the poleward spread of many marine organisms, resulting in the tropicalization of recipient ecosystems. Now, tropicalization is the process by which temperate and subtropical systems become more tropical via the addition of tropical fauna to their assemblages. Now, these tropical fauna can have a wide array of impacts on recipient ecosystems. However, herbivores tend to have the greatest impacts, and that's because they can, one, compete directly with local herbivores for resources such as food and space, and also have great indirect impacts on ecosystems as a whole through trophic cascades by actually feeding directly on foundation species such as seagrasses and macroalgae. So, for example, in southern Japan, as you see on the top left here, where there was once huge, sprawling, beautiful, productive kelp forests, there's now empty barrens, almost devoid of life, because tropical herbivores with warming sea surface temperatures migrated northward into these areas and actually extirpated the foundation species. Now, this is relevant to the northern Indian River Lagoon because we might be experiencing this phenomenon for ourselves. As you can see by this graph, which has mean catch per unit effort on the y-axis and time on the x-axis, sea bream, indicated by the blue line, have increased exponentially since 2017. And they've also expanded significantly northward. Now, I won't go into that because that's covered by Dwayne Edwards in his poster, in the poster session today. These data were taken directly from there. However, you can also see that sheepshead are simultaneously declining in that time period. Now, you obviously can't infer anything from two trend lines. However, we already know that sea bream are negatively impacting sheepshead in the Indian River Lagoon. In 2017, researchers captured individuals that were actually hybrids between these two, which have never been discovered anywhere before. So sheepshead are losing reproductive effort to this dead lineage instead of their own population, and they're also likely competing for resources with these new tropical invaders, if you will. That's because sheepshead of the northern Indian River Lagoon are actually herbivores. Unlike everywhere else in their range where they consume small invertebrates like barnacles, mussels, etc., the sheepshead of the northern Indian River Lagoon consume mostly seagrasses and macroalgae. So what's likely happening with this tropical herbivore is they're coming in, consuming seagrasses and macroalgae, and this is having negative effects via competition on local sheepshead populations, hurting their numbers. This is actually a double whammy for sheepshead because they also rely on seagrasses and algae for recruitment zones. They need seagrasses and vegetation for nursery habitat. Without them, you have fewer recruits, and fewer recruits means less juveniles to replace adults as they die off. So it is potentially creating this positive feedback loop in the wrong direction for sheepshead populations. Furthermore, this mowing down of seagrass beds is likely having community-wide impacts through interaction cascades. Now, I know there's not that much seagrass left in the northern Indian River Lagoon in the first place, but that's also really important to keep in mind because this could be the last straw for seagrasses in the northern Indian River Lagoon. The last thing they need right now is a voracious tropical herbivore coming in and feeding on them. So based on this, my questions are, does the tropical sea bream display dietary overlap with local sheepshead populations? And does this facultative herbivore consume seagrass in the northern Indian River Lagoon? To answer these questions, I worked with Florida Fish and Wildlife Fisheries Independent Monitoring Program in Melbourne, Florida, to collect sheepshead and sea bream from the area surrounding Sebastian Inlet. To do this, we used a 183-meter beach seine net. I then brought these fish back to the lab, excised their stomachs, identified their gut contents down to the lowest possible taxonomic level. I then recorded blotted wet weight and volume of each gut content. I then used the percent mass of each item in the stomachs to run a permanova to detect holistic differences between their diets. And then I used a canonical analysis of principal coordinates and a similarity percentage analysis to tease out what actual items were driving the differences we see. So I had to look at a bunch of disgusting stuff, so now you do too. So here on the left, we have red drift algae taken from a sea bream, a caulerpa rhizome. On the right, you'll see we have green hair algae. Here on the left, we have haliduli, if PowerPoint wants to work. And on the right, you'll see a brittle starfish. Now, visually, I didn't really see a difference. Any of these petri dishes could have came from either fish, visually speaking. However, there was a statistical difference between the species. This is the results of a permanova, which uses a rank similarity matrix to detect statistical difference between communities. In this case, it's the gut content communities of these two fish. So there were holistic differences, but to tease out actual differences, I used this ordination method, canonical analysis of principal coordinates. It's similar to an NMDS, except you can actually feed it a variable that it will then emphasize differences between. So what you need to know about this is the farther away 2 points are, the more different they are. And as you can see, there isn't really much to see. There's a lot of overlap. I mean, in my opinion, there's basically no separation. So it's likely a relative abundance difference rather than an identity difference. So to investigate this further, I use similarity percentages, and on the y-axis you can see I have average proportion of mass for each gut content. On the x-axis I have the gut contents. And what SIMPR does is it actually calculates the percent contribution of each gut content to the differences between diets. So as you can see here, red algae was the largest contributor to differences between the diet. And while it's important, you can obviously see that sea bream ate a lot more red drift algae than sheepshead, but keep in mind that sheepshead also ate more red drift algae than anything else. Second, we have green hair algae. Then we have unidentified mass, which is typical for gut content analysis. And fourth, we have haliduli, which is interesting because even though it's a top contributor to differences between the diets, there really wasn't that much of it in the guts. As you can see, we have about 10% of sea bream diets being dominated by haliduli and about 5% of sheepshead, though it is very variable. So back to my questions. One, does the tropical sea bream display dietary overlap with local sheepshead populations? Yeah, it does. We had a significant difference in the Permanova, but this very powerful ordination method, as well as SIMPR, suggests that there is a lot of overlap. So it's likely a relative abundance difference that's driving the statistical difference rather than an identity difference. They are eating the same things. And two, does this facultative herbivore consume seagrasses in northern IRL? Meh. They do consume them, so it is something to be worried about. However, it wasn't that much. Now, that said, this could be because seagrass abundance is very patchy in the northern Indian River Lagoon. So if it was more abundant, we might see it more in the diets. We'll see. That'll be addressed by future work. So in the future, I want to look at the gut contents of the hybrids I mentioned, see how they're overlapping with their parents and seeing if they're creating competition for sheepshead in the northern Indian River Lagoon. Stable isotope analysis to determine basal resources of these species to get a better picture of are they overlapping in terms of trophic competition and dietary contents, and then spatial analyses to elucidate relationship between capture location and seagrass consumption. Thank you.
Thanks, Adam. Great stuff. One question from the forum. Did you see the microplastics in the guts?
I did not. I didn't see any actually, which is a good thing. It was all natural materials, which is
that is a good thing. Okay, another question then. A series of questions really. Have sheepshead exploited these rates of change over the years? And do you think that the animals are targeting small inverts on the vegetation? In other words, epifauna?
Yeah, that was something I expected going in. However, we really didn't find enough invertebrates, in my opinion, to suggest that these gut contents were absolutely dominated by algae and seagrasses. There were some, like you saw with the brittle that had a whole brittle star or maybe some crab remains. So that would be purposeful ingestion. However, in terms of smaller invertebrates, they pale in comparison to algae and seagrasses in the guts. So that would infer that invertebrates were, just accidentally ingested, or imply that invertebrates were accidentally ingested.
Right. Okay, one last question then. Did you find any sponge in the guts?
Yeah, I think in one or two of the fish there was a little bit of sponge, but not much. Not much.
Well, thanks so much, Ed.
Sure thing.
All right, next up, we have Holly Sweat to tell us about the IRL species inventory.
As I was saying, I'm really happy to be here today to talk to you about some of the continued development that we've been doing on the Indian River Lagoon species inventory. And for those of you who may not know about the inventory and its history, it actually started with a conference at Harbor Branch, the first ever IRL biodiversity conference held back in 1994. And at that time, scientists put their heads together and started cataloging the species in the lagoon system across many different habitats and phyla. A few years after that, the Smithsonian Marine Station became the repository for that catalog. And we added a wealth of information and developed it into an online database and educational tool. And that was 20 years ago. And since that time, the IRL and the world has seen a lot of changes. And so it was long past time for us to renovate the inventory. So in 2019, we were fortunate enough to enter into a renewed partnership with the IRL National Estuary Program that is sponsoring a 5-year renovation and expansion, including moving the inventory to a new domain at irlspecies.org that is actually built around the widely respected and used Symbiota biodiversity data management system. Now, expanding data on the inventory is a never-ending process because we're always going to have new discoveries. And it also takes two components. First, you have to expand the data, and then you also have to expand data accessibility. So I'm going to talk about both of those things today. And I thought with regards to expanding data, it would be useful just to share a couple of examples of projects that we have going on that are generating data that are feeding into this new and improving species inventory. So as many of you know, the Smithsonian Marine Station benthic ecology program has been monitoring infaunal invertebrates in the lagoon since 2005 as indicators of ecosystem condition and environmental change. A new project that we've started in 2020 as a collaboration between the Smithsonian and Harbor Branch researchers is to expand our knowledge of IRL sponge biodiversity, which has been sorely lacking over the years. Both of these projects incorporate morphological identification and also some molecular tools like CO1 and 28S gene barcoding. Over the past 16 years of monitoring infauna, we've collected over 1.2 million individuals, 976 taxa, and over 200 of those taxa are new records for the inventory, which are going to be incorporated moving forward. And I think one of the most useful parts of this data set is the ability to compare distributions of taxa that are variably sensitive to different environmental disturbances. And these data, coupled with other information that we get from the sites and from the species, allows us to start to tell a story about what's happening with regard to ecosystem health at different places in the lagoon, and also what stressors can be affecting those areas more or less. Now, the sponge project and experiments are actually telling us a different story, and a very exciting one. As I said, the original inventory didn't have very many sponge records. We only had 14. Over the years, with some additional projects, we've been able to raise that number to 51. But since we started with this sponge project in the summer of 2020, visiting 25 sites and collecting 261 samples, we've been able to increase that number to 81 taxa, with 11 of our sites having new records for the IRL. And we're not done yet. We expect this number to keep climbing the more samples we collect and analyze. So this is just an example of how little we really know about IRL biodiversity. After 25 plus years, we're still cataloging taxa, and we'll probably continue to do so. So that's just part of the story. As I said, we also care about data accessibility. So if you were to go to irlspecies.org right now, hopefully some of you can do that, this is what you'll see. This is what the homepage looks like. And you'll have a search bar here. You can now search for both common name and scientific name, or you can do an advanced search to generate a list of taxa for certain taxonomic levels. Either way you do it, you have access to these species profiles. We have a little over 500 of our taxa on the inventory with these full profiles. Our ultimate goal would be to have a completed profile for every species listed on the inventory. And hopefully we can do that with a lot of your help. Now, a new thing we're going to be incorporating in 2021 is occurrence data. This has never been on the inventory before. And this is an example of another Symbiota portal that has a functioning occurrence data module. This is an herbarium portal that handles plant records from Arizona and New Mexico. And I just did a quick search for agave collections around the Phoenix area. And I'm able to click on each one of these points and bring up a collection overview with all the information, images, and links to other resources. So if you're used to something like I Did Bio or iNaturalist, you might be asking yourself, how is this any different? Well, the big unique thing about what we're trying to do on the species inventory is that it is geared toward the IRL. So we have the ability to add in a lot of different data types that are useful for folks that are working around here. So for example, after we finish this occurrence data module build out, we'll be adding in other data layers, things like habitat type and coverage, stormwater outfalls, restoration sites. So you can actually overlay those data layers onto your map with the occurrence data for whatever you're looking for. And we think that that could be a really useful tool for everyone. So I realized this was a 20,000 foot flyover of a lot of information in a very short amount of time. But the big take home here is that we really need your help. If you have data that you think could be useful to our effort, please contact me. That includes things like pictures, anecdotes, species information. I invite you to take a look at the inventory and navigate around. And if you have suggestions or questions, please contact me. I want to thank everybody that's been involved with this project. It is a massive effort, and we really appreciate all your help. And then finally, if you're interested in learning more about the infauna or sponge projects that I mentioned here today, I encourage you to check out the talks from my colleagues that are happening later today and tomorrow. That's it for me. Thank you for listening. Hopefully we have time for a question or two.
Great job. Thanks, Holly. Yes, one question. Will you be allowing masking of locality data for endangered species or sought after rare species?
That's a good thought. That's something that I have not considered, but we should definitely talk about that. I would be open to that. Makes a lot of sense.
Very good. Well, keep up the good work. Hopefully someday we'll be able to claim the most diversity in North America once again.
Thanks, Jeff.
Thank you. Next up, we have Danielle Huffner to tell us about the IRL technical report.
So hi, I'm Danielle Huffner. I work for Applied Ecology, and I'm here to talk to you about the progress that we have going on for the State of the Lagoon technical report. For those of you who don't know, we were hired, or not hired, but we were brought on in 2019 by the IRL NEP to kind of go through and put together the first technical report for the lagoon. There hasn't been one, so it's really exciting that we're finally getting the chance to do this. And we brought on MRC to help be our partner with this. And we have a lot of obviously really specific objectives in mind for this, but some of our main or broad goals are to be evaluate some of these key stressors and assess conditions that are currently going on in the lagoon, but also historical trends that have taken place in the past. And then from our information that we've gathered, we can kind of use this to go ahead and look into future stressors and anticipated changes that are going to occur. And we can also use that information to help, find gaps in data or in research or even maybe policy that we could help enact or fill in or find ways that we can fill these gaps. But one of the ultimate goals that we also want to do is provide a document that's going to be able to provide recommendations to the NEP itself and advise their CCMP, or their Comprehensive Conservation and Management Plan. So speaking of the CCMP, this is the vital signs wheel. I'm sure many of you are all very, very familiar with this, but if you're not, this is a list of 32 issues that are either individually or collectively responsible for directly affecting the restoration or the overall health of the lagoon. So when we were thinking about this technical report, we're obviously going to focus on the one lagoon and one community aspects. Those are going to be the bulk of the material. And then the report itself is going to be in this one voice category. And so it's going to be in there because it's going to be communicating the current status of the lagoon and its watershed. So it'll be used for that. And the challenge that we were trying to kind of approach was identifying appropriate indicators for those vital signs and metrics to use in a systems-wide approach. And we looked at a lot of different NEP technical documents from other NEPs, and we found that the Narragansett Bay is really kind of the best example, and so we wanted to follow in its footsteps. And then we also implemented the EPA-approved DIPSR framework, which will help kind of bind all of these indicators together in synchrony, so to speak. And so thinking of this, we came up with this conceptual diagram. And I know this looks like a lot, but this is really what we put together. It's modeled after the Narragansett Bay. They have their own conceptual diagram, but it also brings in that DIPSR aspect of it. So on the left, we have our stressors, so indicators that are representative stressors on the system. On the right, we have conditions of how those stressors are playing out within the system and how those both affect healthy communities. And so this went through a couple iterations, and we have we got comments back from the NEP staff and our technical advisory and steering committees as well. And so I know this is a lot, and I'm not going to get into much more about this, but just know I'm going to focus on the stressors part for right now. If you have questions about it later, you can go ahead and email me. I am free to talk. So this was one of the major things that we did in year one. Was that conceptual diagram. And other things that went on during year one were really kind of putting together, you know, our committees, our table of contents, work plans, things like that, and and some preliminary data digging. But most of it honestly went on during COVID. We've been kind of working from our offices and our home offices, but luckily nothing was really hindered when we did this. We kind of just kept along trucking and didn't really have any problem. But one of the major goals of year one, because it's a multi-year project, I forgot to mention that, sorry. But going into year two, we were really trying to go and hold a science assembly. And as you can probably tell, that just wouldn't work with COVID. What we really wanted to do was have all of the lagoon experts, research scientists, people who know the system best and brainstorm and figure out the best ways to bring this report together and kind of, you know, figure out the best way to organize things. And then after that, we would be able to perform data acquisition and analysis and kind of put some content together. And with COVID, we know that we weren't able to do that. So we tried to hold off as long as we could, but we had to go into working webinars, just like the symposium did. And we had to put together, you know, a quick gist of how to do it, what platforms to use in a short amount of time. But really, since the goal of the science assembly would be to go ahead and figure out, you know, what we need, or we wanted our feedback for the report, and we really just wanted suggestions and ideas. We wanted something that was really analytically driven and polling driven. So we chose GetResponse. And so for our webinars, we did them in late December of this year of 2020. And we went through and we had 2 sessions over 2 days. The first one focused on our landscape and physical stressors, as well as internal and external pollution loading stressors. And our second session was biological and climate change stressors. And you can see some of the things we kind of talked about during those over in the little squares to the right. But the difference between doing a webinar and the science assembly was that we didn't want to waste anybody's time. We know that everybody's doing these meetings. Everyone, now that we're seemingly more available because we don't have to go anywhere, it seems like we have a lot of availability, but it bombards a lot of people. So we wanted to put as much information that would be useful into the smallest amount of time. And so we tried to do a lot of early data analysis and acquisition and be able to present our attendees with options. So we kind of did the foreground of thinking, okay, well, we have this data. How do we want to present it? What do we think it's going to look like? So we would give or get the most out of this. So we would present options in the system and then do polls and get some really good feedback. And it worked really well. It was pretty simple. And that's also one thing we liked about it. It was very user-friendly. And so that leads into where are we now, a couple months later, or less than 2 months later. So all the responses that we've gotten back have been compiled, and we got a lot of feedback. It was so useful. It was great. We've even had a lot of people come up to us and say, we would like to have a follow-up discussion just based on this one area where you're missing some stuff, or we could go into further details that we really couldn't get into in a 2-hour time frame. So that's been wonderful to the people who've been able to reach out to us. Obviously, data analysis is always ongoing. It's going to be ongoing for a little while because it's a multi-year process. So we're trucking along with that. And then the most important part is now we're also looking for interns and peer reviewers. So any students out there who would like to get their hands on some data and get some really great experience and want to help out with this report, shoot me an email. We would love to have you on the team. And we're also looking for peer reviewers too. On top of that, anybody who has a specific field that they obviously are an expert in, they'd like to review the content relevant to that material, or even write some and contribute and become an author on the technical report, we're more than willing to let you help. We would gladly take the help. Anything you guys want to give would be lovely. Or if you have data that you want to contribute and make sure it gets into the report, that would be awesome too. We would love to have it. And with that, I don't know where I am at time, but I just want to say thank you. Thank you to letting me present today. Thank you to the people who've helped with this project thus far, and most notably, thank you to the NEP who funded this project. We are just so floored that we were allowed to do this, and we're very thankful that we get to work on such a meaningful project. And with that, I'll take questions, I believe.
Great job, Danielle. Yeah, you're right on time. I'm not seeing any questions, but if anyone has anyone for Danielle, you can put them in the chat and she'll respond to them. But I'll just remind everyone of her admonition there that they will need some help in terms of review with this document in the future. So please, some of you step up and do that. All right, next up, we have another student talk. So attention judges, and this will be Jacqueline Rumberger talking about multidisciplinary research.
Thank you. Before beginning, we would like to acknowledge that the IRL was part of the ancestral and traditional territory of the Ais and Iroquois tribes of Florida and descendant Seminole Tribe of Florida and Miccosukee Tribe of Indians. We present preliminary results from research conducted by an interdisciplinary team of natural and social scientists. Our collaboration aims to understand changes in the socio-environmental system of the Indian River Lagoon over the past 10,000 years. Long-term goals for the project include documenting geologic, hydrodynamic, and ecological change in the Northern IRL, and evaluating the ways in which human populations have both generated and responded to these changes. We hope results will ultimately provide context for future resource management decisions by extending our understanding of this unique ecosystem into the distant past. This brief overview describes changing environmental conditions and human resource use, particularly in Mosquito Lagoon, before and after the last southern inlet closed around 1,500 years ago. We look forward to your feedback. Also, this figure shows where our data are taken from. The blue triangles represent archaeological sites included in our data set. The yellow star is the location of the last inlet that closed, and the pink square is the location of where the sediment core or vibra core was extracted. This project integrates methods from 6 disciplinary fields. Archaeological data from recent excavations on Cape Canaveral combined with previously reported research allow us to trace long-term changes in human hunting, fishing, and collecting practices. Archaeological fauna can be compared with modern ecological data to identify potential changes in aquatic fish and shellfish communities. Sediments from a geologic vibra core taken in Mosquito Lagoon offer information on changes in trace elements, heavy metals, and when combined with paleoethnobotany, local plant communities over time. The timing of geologic and anthropogenic processes is determined through radiocarbon dating of organics found within the core. Political science research has considered people's resource use choices from the late pre-Hispanic period to the present. Hydrologic modeling enables us to consider how water flows through the Mosquito Lagoon today and how that might vary from past conditions and affect future management choices. Please see tomorrow's presentation by Hernandez and colleagues for more information on this. Vertebrate and invertebrate animal remains from bone and shell were obtained from archaeological excavations at the burn site on the Banana River, shown here, and then which was co-directed by Barber. 3 shell refuse sites around Mosquito Lagoon, an example of one of these is in the top right, and 1 open water site on Cape Canaveral, directed and reported by Dickinson, Horvath, Hellman, and Penders, respectively. The ages of these materials obtained from radiocarbon dating offer a glimpse of what species were living in the IRL at different points in the past. The geologic vibrocore sample shown here in the center is a record of sediment deposition in the lagoon and coastal floor over the past 9,000 years. Phytoliths, microscopic plant silica distinct to family, genus, or species, buried in the core and shown on the bottom right, reflect plant communities over time. We combine these data sources to characterize past ecological conditions at known points in time. Human activity on the east coast of Florida extends back approximately 10,000 BCE, before the IRL was formed. As shown on the timeline, sea level in the southeastern US has been stable since about 4,000 BCE, and the broad ecosystem characteristics of the IRL have changed little since that time. People began intensively using the lagoon's resources around 500 BCE. Despite long-term ecosystemic stability, our data suggests that smaller scale changes may have resulted from inlet openings and closings in response to major storms and the longshore movement of sand. In Mosquito Lagoon, Meta and Brooks have found evidence that 5 natural inlets have opened and closed over the past 6,000 years, with the last inlet closure about 15,000 years ago near Turtle Mound on Cape Canaveral seashore, as shown on the timeline up here. 2 periods of climatic warming here and here fall just before and after this last inlet closure. The bulk of our archaeological, geological, and paleobotanical data date to this period, right before and after this inlet closure and during these warming periods. This allows us to analyze the immediate and then longer term effects of this ecosystem change. Phytoliths from the vibrocore show a change in plant populations around Mosquito Lagoon after the Turtle Mound inlet closure. The graph demonstrates an increase of arboreal phytoliths, predominantly palms, from pre-inlet closure to the present, along with a decrease in phytoliths from grasses. Archaeological evidence from sites shown here on the map indicate changes in exploited aquatic species following the inlet closure, although data are insufficient at this time to demonstrate a causal relationship. Leading up to 750 CE, eastern oysters and to a lesser extent saltwater clam species were exploited, while later, approximately 200 years after the Turtle Mound inlet closure, indigenous people emphasized saltwater clams, specifically coquina, and to a lesser extent saltwater mussel. As you can see here on the table, reliance on saltwater clams remains pretty stable. However, the dominant species of clam exploited shifts. The emphasis on coquina and mussel over the past millennia is also evident on the Banana River at the burn site, where inlet closures should have not affected local conditions. In terms of exploited finfish, various species of jacks, catfish, and sheepshead were more commonly exploited prior to 750 CE, while mullet, catfish, sea trout, and drum species were more commonly exploited after. We are currently evaluating whether seasonal changes in human resource use, overexploitation, varying food preferences, or technological changes could explain these shifts as opposed to fluctuating ecosystem conditions. Recent changes in the northern IRL ecosystem conditions are related to colonial, federal, and industrial era activities. The figure on the left shows the increases in lead, zinc, and copper by a factor of nearly 6 over the past 200 years, likely due to industrialization and agriculture. On the other hand, as you can see in the figure on the right, sodium, which is in blue, gradually decreases by 29%, perhaps as a result of the closure of the Turtle Mound inlet. The IRL is not only an ecosystem, but also a social system. Political science analysis has focused on defining technologies and institutions for resource management and extraction over the past 700 years. Economic organization has changed from a subsistence focus prior to 1565 to mercantile capitalism and finally modern market capitalism. The priorities of living people within these political economic regimes varied, increasing pressure on the IRL's natural system. Hellman has proposed a slow overexploitation of clams, evident by reductions in individual size and age starting 1,500 years ago. This 1,000-year process also began with a shift away from oyster exploitation. Changes under capitalism were accelerated with possible fishery collapses near St. Augustine and the early colonial period, and around the IRL, agriculture and residential land use rose by factors of 2 and 5 between 1943 and 1990. While much of the work remains, we see preliminary patterns in these data. Despite broad ecosystem stability, changes caused by adjustments to water exchange with the Atlantic may have had gradual but meaningful impacts on both fauna and flora within the lagoon. In the Mosquito Lagoon, decline in the oyster population around 750 CE. It is possible that by 1,000 years ago, human exploitation of oysters was affecting species availability. We presume that resource depletion has accelerated with the advent of capitalist political economies, although we lack archaeological data for the past 400 years to quantify impacts. These results provide a starting point for the next phase of our research that will use a coupled food web and hydrodynamic model from deep time to present. The model will simulate how lagoon floral and faunal communities responded to changing hydrodynamic conditions. This modeling suite will not only reconstruct the food web starting 1,500 years ago, but could ultimately be used to simulate effects of modern proposed inlet openings or other changes to flow in the system. In the development of these models, we will include information on how humans have responded to and caused ecological changes while considering the longer time scales at which processes play out within the IRL system. Thank you.
Jacqueline, thank you very much. Fascinating.
Thank you.
Not seeing any questions in the chat box. Oh, wait, here comes one from Annie. How do the past periods of climatic warming compare to existing conditions?
There has been increased warming since the Industrial Revolution, although there was a small period, what's called a little ice age, between the last warming period and the Industrial Revolution, but it's unclear whether that has had effects in this region.
Okay, thanks so much. All right, lastly in this session, we have Michelle Gaither. She's going to tell DNA biomonitoring.
All right, so I'm going to give probably the most brief overview of environmental DNA that can be given, 10 minutes is there's a lot to talk about. So I'm going to start by describing or first defining what eDNA is. Okay, so broadly defined, environmental DNA is any genetic material that is shed or excreted from organisms. It can be skin or hair, feces, gametes into the environment. And in this case, the environment can be sediment, it can be soil, it can be water, it can be even air. Now, since we are a marine lab, we collect water samples, okay? And so our water samples include not just material that's excreted from multicellular organisms, but also whole microscopic organisms, okay? And we include that in our definition of eDNA as well. So what do we do with those water samples once we have them? So we have to be able to isolate the DNA from our samples. So we use filtration, which is what most labs do. And then we chop up our filters into small little bits, and we use any of a number of commercial kits to extract our DNA or some in-house formulation, right? So now we have our DNA isolated from our water samples. And so what are the next steps, right? And there's a couple of different assays that can be used. You can either use a single species detection approach, or you can use a metabarcoding approach. So for a single species detection approach, this is focused around designing species-specific primers that are designed to amplify your target, your species of interest, at the exclusion of all others. Now that might be an invasive species, or it might be something that's rare and endangered, or something that's particularly ecologically important. This process of designing these assays is actually quite intensive, and so it would be a species of particular importance. And then you can apply a qPCR or a ddPCR, which is digital droplet based approach, to run your assays and determine whether or not that particular target DNA was found in your DNA. Okay, in your water samples. Okay, now there are lots of published assays out there. The freshwater folks really sort of set the stage. There's a lot of published assays out there on an invasive species in freshwater, but there's also assays that have been published on things that are probably a little closer to our interests, like small tooth sawfishes, bull sharks, etc. In our lab, we've been working on a couple of different single or species-specific approaches. My postdoc, Girish Kumar, who you'll see his name a lot during this talk, he's really been the one who has worked hard to establish protocols in my lab. He's working with FWC to on red tide, on a red tide assay, a ddPCR. Undergrads in my lab have been working on qPCR approaches to look at lionfishes and dwarf seahorses. Potentially we can use in the IRL as well. Now, the bulk of the work in my lab is focused on metabarcoding approaches. So instead of designing primers that amplify only one taxa at a time, these approaches are designing primer sets that will amplify an entire group. Now that group can be all metazoans at the broadest level, or it can be just species within a specific genus, right? So you apply these designed primers, these newly designed primers, to your samples. You create these libraries, and then you sequence them on a high throughput sequencing platform like the Illumina MySeq. Those platforms will generate millions of reads that you then have to bioinformatically pull apart, right, and then assign to some, to a taxonomy. Now, an example of this kind of work is this paper that was published a couple years ago. Now, in this case, they apply eDNA techniques, and they found 13 taxa, right? But they also looked at visual census data. They looked at BREV data, which is baited cameras. And what they found was that they had 44% more sharks were detected using eDNA. And the reason why I highlight this particular project is because in most of the studies that you look at where they compare traditional techniques with environmental DNA techniques, you typically get more species with environmental DNA, but no single method can fully characterize the community, right? There's no single method that seems to be able to detect all taxa. And so these combined approaches are usually what's recommended. Now that was also true for the work that we have done so far in the IRL. So this is, we're comparing here eDNA data with trawl and seine data. The trawl and seine data we got from Jeff Cook's lab here at UCF with the help of Dakota Lewis, who's a student of his. So thanks to both of those folks. So here, if you look at, here's a species list, which you're not meant to read. The trawl and seine data picked up 46 species, right? But the eDNA data that in the same area, roughly around the same time, picked up 76 taxa. And if you look at the Venn diagram, there were 36 that overlap between the two methods. There were 10 that were picked up only in the nets, and 40 that we picked up with eDNA that they didn't find in the trawls and the seines. So what kind of species are we picking up in those? The eDNA sample, right? Our analyses versus the trawls and seines. We're picking up sometimes a lot of small species, like we got mollies, right? We got more gobies, but some other taxa as well. And we're getting better taxonomic resolution. And this is an example, just look at the family Calerpidae, right? So looking at the eDNA data, we resolved 5 species. They resolved 2 taxa at a species level, and then it was the menhadings that they collapsed to genus, and I assume that's because they're taxonomically difficult to distinguish. But without any kind of taxonomic expertise, we were able to pull apart, those 2 menhadens. So our increased number of species is multi, it's multi-pronged. We're getting some of the smaller stuff that's probably avoiding nets, but we're also just getting better resolution in some of these hard to pull apart groups. Now this second data set, this is part of a much bigger methods paper that we're looking at. So when we first started optimizing eDNA protocols in our labs, we wanted to know whether or not we needed to treat samples differently if we are sampling in the IRL versus outside. So we have 2 sample sites, one inside the lagoon and one outside of Sebastian Inlet, okay, just outside of the plume. And so we tested in this, we tested extraction methods, pore size for filters. We also tested or looked for PCR inhibition. And this is just this is the results from inside the lagoon. Now, this are the different extraction methods that we tested out. That's not what's important, my point here. But what we did find, we got lots of DNA in these samples from inside the lagoon. We got lots of PCR inhibition as well. We detected 36 species. The bait anchovy was dominant, okay? What was interesting though is when we compared that with the samples from outside the lagoon and that clear, what we characterize as clear water sites, right? We got way more variability across our replicates. First of all, we got far less DNA in those water samples. We got far less PCR inhibition, which of course was a bonus. But we also got way more variability across our replicates. We ended up detecting 37 species in those samples, with the Atlantic threadfin herring being the threadfin herring being the dominant species. But the take-home message really from this project, at least for our research purposes, is that in these more open ocean environments, that much more extensive water sampling would be required if we really wanted to characterize those communities, because it seems as if the DNA is sort of haphazardly distributed, and our water sample sizes were only 500 mils at the time. So now, the big question everybody wants to know is, can eDNA be used to estimate biomass? And the answer is, well, probably, but maybe. It's going to be complicated, okay? So if you look at study after study, you'll see there's often a correlation between, and sometimes better than others, a correlation between eDNA concentration or read counts and, biomass or individual counts, okay? In fact, there's a review paper that's going to be coming out soon. We're working on a special issue in the journal Environmental DNA on marine metabarcoding, right? This review paper looked at 55 examples. 93% of them had a positive correlation between eDNA concentration and biomass. So the correlation is there, but there's a lot. What makes it complicated is the fact that there's a lot of variables. So DNA shedding rates, for instance, vary significantly across species, but also across life stage, body size, etc. DNA residency times is going to vary depending upon bacterial communities, UV exposure, etc. So a model that might be fine-tuned for, walleye up in Wisconsin can't be applied to anchovies, in the IRL. And each one of these models is have to be adapted for the specific location and probably the taxa of interest. So my student, Emily Farrell, is a PhD student here at UCF, is going down this road. She's working with Rich Paperno's group and collecting water samples along with the trawl surveys that FWC does in the Northern IRL. So hopefully by next year, we'll have something to report there. So lastly,
you're there. Very good. Keep going.
Okay, last slide. I'm almost there. So speaking of a taste of research during the pandemic, I just wanted to highlight this work that we did last August, July, when all of our other research possibilities had sort of been canceled because of the pandemic. Myself and my 2 PhD students had spread out across the state and sampled across 11 different locations. Again, 540 sample locations. We're really looking at biogeographic patterns at a larger scale, at least the statewide scale, with this project. And again, hopefully we'll have something for you guys next year. So with that, I have no idea how I did on time. I either talked really fast, who knows? I need to thank a lot of folks here at UCF and elsewhere, FWC and the Kennedy Space Center Ecological Program, funding sources, etc. And with that, anybody has any specific questions? Because I know we don't have much time. There's my email address.
Thank you, Michelle. There's numerous questions in the chat box, so I'll ask you to deal with those as best you can today. And of course, you guys can always email her as well. Thanks so much to our speakers. Great first session. I'm going to turn it back over to Dennis now.
Hello, everyone. This is Irene Arpajoglu, and we're going to begin with oral session 2, Indian River Lagoon Restoration. Our first presentation is a student presentation, so any judges out there, please pay attention. This presentation is Restoration Methods for a Successful Mangrove Living Shoreline in Mosquito Lagoon, presented by Rebecca Fillay.
Hello, everyone. As my title suggests, the overall goal of my research is to increase the success of reservoir mangrove living shorelines. Now, living shoreline utilizes natural elements like vegetation and oyster shells as a shoreline. Specifically, I'll be focusing on living shorelines comprised of red mangroves. Previous research has shown that mangrove living shorelines can effectively reverse patterns of erosion while also providing native habitat. The one caveat is that living shorelines can only be successful if the vegetation survives and grows. So I asked 2 questions. First of all, how does starting mangrove age and presence of a breakwater employed mangrove mortality? So I hypothesized that breakwaters would increase survival via reduction of wave energy, And also, older deployed mangroves were hypothesized to survive better due to their increased ability to withstand wave energy. So to test these hypotheses, an experimental living shoreline was deployed along 650 meters of intertidal shoreline in Mosquito Lagoon. This site is a shell and clam shells, in addition to other items such as broken pottery. This makes the sediment very shelly and can make it difficult for mangrove propagules to recruit. So recruited adult red mangroves and several scarps were present, such as the one pictured here on the left, which is an indicator of erosion. To start off, red mangrove propagules were collected from Mosquito Lagoon. So mangroves at 11 months were classified as seedlings, and they were easily identified by their soft, herbaceous stems that didn't have any woody cortex. And then transitionals were 23 months old, and they were named so because they started to develop their woody cortex, and it covered about 25 to 75% of the stem. And then finally, adults were between 35 and 47 months old, and they had a fully developed woody cortex. So we took measurements for each mangrove. Measurements included height, diameter, the number of branches, the number of leaves, and then we had grounded prop roots. So if you look at this picture, a grounded prop root is a secondary root that starts at the stem and reaches down to the sediment, whereas a free hanging prop root stem, it's at least 2 centimeters long but hasn't reached the sediment yet. And based on a NOVA test, each measurement was significantly different among the developmental stages. 6 treatments were deployed along the restoration site, made up of each developmental stage, both with treatment was randomly determined prior to the restoration. Within each treatment, 16 mangroves were placed in 2 staggered rows of 8 at an elevation inhabited by the nearest naturally recruited adult red mangrove. Each breakwater unit was made up of 2 rows of 21 oyster bags, and each treatment was 6.6 meters long, and it was seconded by other treatments by 6 meters as well. A number tag weighing approximately 4 grams was attached to the mangroves with flagging tape for easy identification. The flagging tape was loosely enough to allow for growth, and the weight of the tag did not cause to bend. Survival was monitored monthly between June 2019 and June 2020, and survival categories include alive, pictured here on the left, standing dead where the mangrove is still rooted in the ground and standing, but it has no green foliage, and then missing where the mangrove is no longer where you put it. So temporal factors were monitored to help identify the reason for mangrove mortalities. Mean water level for each month was identified with 5 wildlife cameras pointed at 5 PVC pipes along the site, such as this one, with colored zip ties spaced 2 centimeters apart. The cameras were set to take a short video every 30 minutes throughout the day for 5 days each month. If waves were present, salinity was taken with a refractometer when the site was visited, and minimum temperatures, wind speeds, and precipitation data were accessed online from the nearest station. So moving on to the results of the experiment, a logistic regression indicated that developmental stage significantly impacted survival, and breakwater presence increased survival, which can be visualized in this bar graph. So results of the regression indicated that breakwater presence increased the odds of survival by 366%. Age increased by 130. Adults increased the odds by 1,053%. So results of a logistic regression ran on total mangrove mortality for each month indicated that water level was a significant temporal predictor, indicating that flooding stress was the source of mortality for the majority of mangroves. Restoration site experiences a season during the fall. You can clearly see this change in these pictures of the same area during January versus September. if we look at this graph showing for each treatment over the 12-month period, 62% of all the mortalities took place in October, when the mean water level was shown by this gray bar for 2 months. So this points to the importance of not only magnitude, but duration of flooding, because although the water level was high in September, it wasn't until we observed a sharp decrease. So flooding stress is also supported by the type of mangrove mortalities. If forceful removal was the culprit, then mangroves would have gone straight from alive to missing, but 93% of the plants were standing dead at some point over the 12-month period. So using the information gleaned from this experiment, we were able to make predictive models from the results of the logistic regressions, looking at the impact of size metrics at the start of the experiment on mangrove survival. So for mangroves with a breakwater, starting diameter and the number of grounded crop roots were significant predictors of survival. And if we interpret this model below, you can see that diameter is on the x-axis and survival probability on the y, and the interaction between the two is changed by the number of grounded crop roots, which is the coloration change. So if we go to diameter at 2.5 centimeters, survival is above 75% no matter what. But then if we add about 3 grounded crop roots, survival is very close to 100%. Whereas mangroves without a breakwater, it was starting diameter and starting significant predictors of survival. Now, the fact that height is important for survival, but only for mangroves that didn't have a suggests that the breakwater presence was increasing survival may have been partly due to the reducing wave height experienced by the mangroves. So if we interpret this graph, we see that diameter and survival are in the same place, but now the coloration change is based on height. So at 2.5 centimeters, survival probability is widely variable based on that height. So if we go 2.5 centimeters diameter and about 80 centimeters for height, then we're just reaching over 75 survival. All right, to summarize and leave everyone with some practical takeaways, if your potential living shoreline site header season, or if you're planning for unpredictable flooded conditions, first of all, use a breakwater. It will significantly increase survival of your mangroves through the reduction of wave energy and lowering of wave heights. Secondly, utilize adult mangroves. Compared to young 11-month-old seedlings, they increase the odds of survival by over 1,000%, and they are easily identified by their stems, which are covered completely by woody cortex. And finally, if you want to further increase your probability of survival among the adult mangroves, choose those with a diameter of 2.5 centimeters or greater, which have already developed a few grounded crop roots, such as this mangrove pictured here on the right. I want to say a big thank you to C-Lab and all the other organizations and volunteers that helped with the restoration and monitoring of the site, because it required a lot of field work. And of course, thank you to the National Park Service for funding the restoration in the first place. And any questions?
You have one question. It is, can you define a standard mangrove tree?
Can I define a standard mangrove tree?
Yes.
Yeah, so mangroves are defined for their ability to zone where it has water. So they have secondary roots that have special systems that are able to filter out that salt. And so it's basically just where they're able to live. But specifically, I was looking at the red mangrove, which is Rhizophora mangle.
Okay, and you did great on time. I don't see any more questions. If anyone does have any more questions for Rebecca, please type them into the chat, and Rebecca, you can just keep an eye on that.
Okay, thank you.
Our next presentation is by David Cannon, Canopy Flow Interactions in Mosquito Lagoon, a review of recent work on eastern oyster reefs and shallow shoal grasses.
All right, can everybody hear me? Hopefully.
Yes, we can hear you.
All right, great. Hey everybody, thanks for tuning in today. My name is David Cannon and I'm a postdoc studying hydrodynamics in North Indian River Lagoon at the University of Central Florida. Today I'm here to talk about canopy flow interactions in the lagoon and give a brief overview of our recent work on intertidal oyster reefs and hopefully seagrass fringes. Don't hold your breath on the seagrass fringes, like a little bit overcommitted with that abstract. So let's get started.
We can't hear you if you're speaking.
Okay, can you hear me now?
Yes.
Frustrating. I think most of you are already on my side when I say that oyster reefs are really important. So they stabilize sediments, promote biodiversity, and improve water quality. Unfortunately, there's been a significant decline in reef ecosystems worldwide over the last several decades. Today, a lot of effort is being made to restore oyster reefs and their associated ecosystem services around the world, including here in Indian River Lagoon. The hydrodynamic influence of oyster reefs changes dramatically depending on the health of the reef. So natural, intact reefs have very complex three-dimensional structures. As water flows over the reef during high tide, velocities are heavily attenuated near the bed within the canopy, and mixing is influenced by interactions with the individual oyster clusters. Above the canopy, the oyster clusters act like large roughness elements, exerting drag on the flow and elevating turbulence at the canopy surface. Degraded reefs, or reefs where oysters have largely died off or have been removed, look very different. So they're largely composed of disarticulated oyster shells, which pile up and create these high, unstable reef crests. Crest elevations are high enough that the reefs are rarely submerged, and the lower roughness associated with the shells leads to weaker mixing. When we restore reefs, our goal is to take a degraded reef and get it to look and behave more like a natural, intact reef. Linda Walters' group here at UCF, who we worked with on this project, really excels at that. So they start by grading the reef to a lower elevation, then they anchor oyster mats with vertically oriented oyster shells to the reef, and they allow live oysters to recruit naturally. That takes a lot of work. They've seen a lot of success with oyster recruitment, but there's still these nagging questions about the hydrodynamic influence of the restored reefs. Namely, do the restored reefs behave like degraded reefs or like natural intact oyster reefs? And do these behaviors change with time since restoration? To answer those questions, we conducted dozens of field experiments on restored and reference reefs in Mosquito Lagoon. The reefs were chosen such that they represent what I'm calling a restoration continuum, including degraded reefs, intact reference reefs, and reefs of varying restoration ages or time since restoration, ranging from 6 months to 4 years. So over this presentation, I'm going to be talking about restored reefs with this convention where they're named R dash and then the year that they were restored in. So the most recent year is also the most recent restoration. We also monitored a single reef both pre and post restoration, with monitoring continuing for nearly 3 years after the restoration took place. So I'm going to be focusing primarily on data collected during this Space for Time study, but I'll mention the other reef briefly at the end of the presentation. So during each experiment, velocity measurements were collected in the main channel and on the reef surface for 3 to 5 hours over a single tidal cycle. A weather station was used to record wave heights, wind speeds, and water levels during all the experiments, and the reef surface was characterized using both a GPS surveyor to get the elevation and a laser scanner, which gave us really high resolution measurements of the oyster canopy across the reef. Importantly, velocity measurements were collected both above the oyster canopy and within the oyster canopy, which allows us to look at differences in turbulence and flow attenuation related to that canopy position. That alone is pretty unique and worth emphasizing because there's actually not many studies that have looked at near bed mixing within an oyster canopy in the field. So that's pretty neat. Let's get to the results. We're gonna start off looking at the structure of each reef. This plot shows the elevation profiles for the 5 reefs included in the SFT study. The red line represents the degraded reef. The green line shows the reference intact reef. The other colors are all restored reefs. So the big takeaway here is that the degraded reef crest, this red line, was significantly higher than any of the live reefs. In fact, the crest of the degraded reef was actually higher than the maximum water level, which might be hard for you to see. It's around here. That was measured on any day of sampling. So water never passed over the reef surface while we were there. This led to some interesting flow behavior, which we'll get to shortly. I'll also point out the restored and reference reefs were fairly similar, with wide crests and lower slopes, although it's worth noting that the live reference reef was significantly wider than the others, and that's just due to the longer sustained oyster recruitment that occurred at the site. Laser scans were used to characterize the surface of the reference and restored reefs. I'm presenting the solid volume fraction as an analog for the roughness. It represents the fraction of space occupied by oyster clusters on each reef and is computed in 50 centimeter grids. I was surprised by how heterogeneous the reef surfaces were. Cluster densities were really patchy across the reef, and solid volume fractions range from 0 to 25%. So that patchiness is hard to quantify, but it's worth pointing out because I actually think it had pretty dramatic consequences on the near bed flow. All right, now that you know what the reefs look like, we can talk about hydrodynamics. At each reef, we estimated current attenuation as the percent reduction in current speeds from the channel to the reef crest. Basically, the higher the attenuation rate, the better the reef was at attenuating energy and potentially protecting the bed and the shoreline. In this figure, the channel speed is on the x-axis and the reef speed is on the y-axis. So currents were strongly attenuated near the bed on the live, reference, and restored reefs, with attenuation rates between 84 and 97%. Remember, these measurements represent velocities inside the oyster canopy, where the speed reductions are caused by both oyster clusters and the proximity to the bed. Differences were statistically significant between the individual reefs, but they weren't linked to live oyster density or solid volume fraction. Instead, we're guessing that the variability was driven by spatial heterogeneity on the reef surface. So velocities above the dead reef were essentially decoupled from velocities in the main channel. There just wasn't a significant relationship. That's why there's no line here. So the lack of submergence meant that the dead reef acted more like a breakwater. Waves ran up and down the reef face, and flow was steered and stagnated near the surface. There are just characteristically different flows there. Above the canopy, velocity attenuation was much weaker, mean estimates between 51 and 65%. Currents in that region were significantly less affected by the bed, so the decreased attenuation rates weren't hugely surprising. What was surprising was the similarity between the estimates of the different reefs. So they were mostly statistically similar, with overlapping 95% confidence intervals, and we attributed those similarities to the fairly constant oyster canopy height across the sample locations, since the oyster canopy acts more or less like a large single roughness element outside the very near bed region. So on to turbulence. This figure shows the vertical turbulent energy and the turbulent kinetic energy dissipation estimated over each experiment. Near bed experiments are shown in red, above canopy experiments are shown in blue. Understanding exactly what dissipation or turbulent energy are is less important than understanding that higher values correspond with enhanced mixing and elevated bed stresses. Near bed turbulence was significantly weaker and more variable within the oyster canopy than above the canopy, with results suggesting, well, the results kind of mirrored the observations of the current speed and attenuation rates we described a few moments ago. If you compare the near bed measurements of the live and degraded reefs, we can see that the turbulence was significantly higher within the oyster canopy than above the dead reef, suggesting that the canopy-induced mixing played a significant role in near bed turbulence budget. Perhaps most importantly, the turbulence rates both near the bed and above the canopy were fairly similar across the live, reference, and restored reefs. A lot of the differences in turbulence can be attributed to differences in the mean velocity at each reef. So you can normalize by the velocity to kind of get rid of that. Values near the bed varied by an order of magnitude, but they are always higher in live oyster canopies than above the degraded reef. Above the canopy, values more or less converged with similar behavior at the reference and restored reefs, but it's important to point out that there was actually a slight trend with restoration age for these above canopy measurements. So if we looked at the reef that we actually tracked over 3 years, a single reef, then we can see that we saw the same type of thing. So at 6 months, the restored reef was very low, or had lower mixing compared to the reference reef, but by 30 months, it had met or exceeded the mixing rate. So what did we learn as I run out of time? Turbulence and mean flow characteristics vary spatially with elevation and with reef condition. Near bed turbulence and attenuation was much greater at the live reefs than at the degraded reef, and near bed flow was highly variable, which I'm attributing to spatial heterogeneity as kind of a grab all. The big important takeaway though, is that the restored reefs were hydrodynamically similar to the reference reef within 1 year of restoration. So the above canopy conditions took a little bit more time to mature, but they were within 50% of that reference value, even after just a year. So the takeaway is that good restoration strategies lead to quick canopy development and hydrodynamic success. So if you take one thing away from here, just know that the reefs will still behave like a reference live reef after they're restored, as long as you restore them correctly. And that's it. Thank you.
Thank you for that, David. There are one comment and two questions in the chat. The first comment is by Dr. Kazemi. My suggestion is to normalize the turbulent kinetic energy with the upstream velocity. And then his question is, have you considered the eddy viscosity in the canopy flow?
No, that's a good question. So I'm guessing that you're getting at, is it low enough? I'm not 100% sure what you're getting at. Maybe you're looking at laminar flow and kind of this increase in turbulence as viscosity becomes more important. We've thought about it a little bit, but the measurements we've collected so far aren't too helpful for determining that. We're developing some new experiments soon where we'll be looking more at that type of thing.
Okay, there are two more questions in the chat if you want to answer them directly in there, since we're out of time and need to move to the next presentation. Next presentation is Todd Osborne, Leveraging Natural Selection for Restoration of Clam Populations and Water Quality in the Indian River Lagoon.
Here, and first to say thank you very much for the opportunity to all the organizers for putting this together, even though we were all kind of separate via COVID. It's a great opportunity to present. I want to thank, of course, my co-authors here, Jose Nunez, who is the proverbial clam whisperer here at Whitney Lab. Of course, Jeff Beal, who has been very supportive of this project from the get-go, and some of the other collaborators that have been working with me lately, Lorraine Simpson from FOS, and then the rest of my staff here, Anna, Sabrina, and Norm, who have been instrumental in the legwork of making this happen. However, I cannot say enough that I did not get my abstract together fast enough to get permissions from everyone that it should be a co-author here. So I listed them in the thank yous. I'm going to thank them first. And that is all the folks you see on the screen here have been instrumental in this process, contributing not only their time and expertise, but also financially in terms of support and or matching funds. And we couldn't do it without them. It's been a very big collaborative process to date. I want to say really quickly that this figure alone probably represents the most influential aspect of what we started to do. And I've been watching all the amazing work going on very closely in the IRL with oysters. And we even modeled some of the work we do up here at Matanzas River based on what was being done down there. And when I saw this figure a couple years ago, I thought, wow, what's going on with clams? And, you know, it's impossible to say that the loss of these particular clams have induced some of the problems we see in the IRL, but I'm confident in saying that they definitely have a role in filter feeding and helping us out with the environmental problems we have right now. And so I'm going to show you a quick video. Yes, I know this is risky in an 8-minute talk, but I want to show you guys just a little bit of what goes on. There's been a lot of questions about this and maybe some misconceptions. First off, adult clams do bury. That was a question I got in several other talks recently, so we made a video to show you. They're pretty adept at doing that, and once we plant them in the field, they bury themselves in a few minutes. And of course, they are very effective at filtering that you'll see here. But the point that I want to make is not that they're acquiring so much of the algae and nutrients that they're filtering, but they are condensing that material into larger particles that settle out. And you see them raining down on your screen right now. Those are pseudofeces, but they are clearing algae and making these particles out of it that then meet the sediment surface. And that's a really important factor in terms of providing that water clarity that we're looking for so much, right, to get our seagrasses back. So that kind of initiated our ideas about clam restoration in the IRL. And the objective, of course, was to improve water quality and clarity, right? And first by reducing algal turbidity, but also by reducing nutrients in the long run by getting these things out as particles into the sediment. We call that benthic coupling, and it's a super important process, something that we want to work more with. And our approach was kind of high risk too. It was to locate, spawn, and repatriate these this variety, we call it, this genetic line of Mercenaria clams. This is the northern quahog, to optimize survival and reproduction in the IRL. And we're hoping that initiating this will spark the activity that follows, which is population growth. And a couple of hypotheses we were working with was just a very simple one. First, commercial aquaculture techniques would be suitable for doing this and would help us out a lot. And we don't have to reinvent the wheel there. But our second would be that the traits selected for by the harsh environmental conditions in the IRL in the last 8 or 9 years could be leveraged for optimizing restoration success. And that's what we really want to get into with the clams. Now, how we do this? Well, we went out and collected broodstock. And there were a lot of naysayers that said, well, good luck, there's no clams in the IRL anymore. And then it took about 500 man hours to locate 39 adult Mercenaria clams. But we took them home, we spawned them, and did they spawn? We ended up with 40 million veligers out of those 39 individuals. And we call them super clams because they really have been through the ringer, and for them to survive what they've been through, they have some pretty hardy genetic stock, right? So we spawn them in the laboratory to the veliger stage, and once they develop their shells, we get them out into our nursery facility where we bring them up. The process is pretty straightforward. We're pumping river water from the Matanzas River up here in St. Augustine through our system, and we grow them in a protective manner until they're big enough to outplant. So you see in the top left corner, seed clams and a couple of millimeters in size. We bag them, we bottom plant them, and we let them grow. And we want to get them big enough to overcome major predation when we get them to the IRL. So we unbag them, sort them by size, and then that size you see in the bottom left is about the size that we go to outplant with, about 15 millimeters across. Of course, the next step is prepping the aquaculture leases because these were found in and around aquaculture areas. We decided that they need to go back into aquaculture leases first. That's an agreement with FDACS who manages that. And so this is a picture from River Rocks down in Rockledge, and a bunch of our supporters and collaborators were there for an outplant this past summer. In the midst of COVID, people were anxious to get outside and do something, and here we planted about 300,000 clams that day. And you can see the PVC markers are marking the sites, right? And that repatriation process is not only a lot of fun, it's hands-on, but a lot of people can do it because we're literally taking clams out and sewing them on the bottom. We utilized 5 major sites this past year, including the Southern Mosquito all the way down to Sebastian Inlet, leases from partners that were critical to outplanting these guys. And you can see here, this is one of our deliveries of clams, and they're bagged up and ready to go. We move them to boats, we move them to the site, and we disperse them. And then we cover them with netting. And what you can see on the top right corner here is clams that have been planted, and just a few minutes later, we take a little cover net over them to protect them from predation for the first 12 months of their time in the IRL, right? And if you look at the bottom right hand, that's a aerial picture from Jose's kite camera, and you can see the plots starting to darken. So those are 14-foot by 50-foot plots that are, can have anywhere from 10,000 to 100,000 clams underneath them. And we plant them in various densities for research purposes. But to date, after our first year, which was October of 2019 to October of 2020, we planted 1.2 million clams in the Southern Mosquito at 2 sites, 2 aquaculture leases owned by a partner, Mike Sullivan from Premium Seafood. And then at River Rocks, we put in 800,000, another 500,000 in Titusville with Sea Grant, and another 500,000 at Grant, where it's Grant Sebastian Lease Complex down on Blair Wiggins lease. So results, well, we're seeing that we anticipated faster growth in some sites due to the algal bloom that we had this past summer. We noticed those growth rates stopped off, but we didn't see a lot of mortality. In fact, we were very, very happy to see on Tuesday of this week that the average size at Cedar Creek, which I couldn't squeeze on the graph this week, is 47 millimeters now. So good growth occurred there. And we're looking at around 96% survival at these sites. This is an aerial view of Turner Flats. These are not all our clams, but this is kind of what a site looks like when it's fully planted. Also, another one that I wanted to highlight was River Rocks, because it's so accessible by the public and very easy to walk out and see. We did note that we had general slowing of growth over the summer, which is expected, and then just a recent data that came back is that things are growing again. We lost a few more clams at River Rocks due to the impressive rainfall we had this past year, an additional 17 inches on top of our normal rainfall. And so salinity problems were probably a major factor, as well as predation from crown conch. We had a crown conch invasion. If you'd like to participate and help us clear crown conch, Blair Wiggins put a great menu item on his website, how to cook crown conch. I'm not a big fan, but it's something that came out of our work. Just note the picture here on the right, there is a little bit of growth on these, and you can see where we planted them out and what they've been doing. So we're very happy about the results there. We did some of our own research in the lab, determining filtration rates in-house, and we picked out about 10 gallons per day per adult clam. And so just looking at that, 30 million gallons per day filtered by the clams that are in the system now. And then their numbers for storage of nutrients is pretty impressive too. About 270 kilograms of nitrogen stored, 90 kilograms of phosphorus, and about 8,300 kilograms of carbon. And so when we think about that process, we modeled it out just to look at what would 5 years look like of the 3 million we put in, or 2.35 when I did this work last year. And you can look at the numbers as they grow. And that's a very conservative estimate of a 10-fold increase in population, right? Because a single clam can produce 1 to 2 million eggs. And with high density planting, it's not uncommon to have tremendous recruitment there. But in the end, 3.5 billion gallons of water filtered over the process is pretty impressive. And that's where we're headed. We really want to see these populations come back. Our preliminary conclusions, of course, are that the gear in the process works pretty well. We don't need to reinvent the wheel there. And with our survivorship, we think we're doing it right so far, even with the constraints we've had. Finally, the clams we put out last year are ready to reproduce already. And we've seen that, and we're anticipating that this spring, which makes for a big deal. Our future plans include engaging commercial aquaculture more to help us with this problem. A variety that would work for them could also be economically advantageous. And so we're pulling that together. And we've got some other things planned as well. Our big shot is the 8 million clams we want to put out with NEP and Brevard Zoo this coming year, and making some inroads with co-planting experiments with seagrasses. Okay, I think I did it in time. Do I get to answer a question or two?
You have 2 questions there.
All right.
One from Stacy. What is the baseline current populations based on?
The baseline of what we're seeing in the system? Well, we could not find them. In fact, the baseline population was almost impossible to determine. Simply put, about 500 man hours of scratching around in former high density places based on previous records showed us that they just weren't there. And that was a bigger problem. And the spatial diversity in where we located those adults was also a hindrance to reproduction, meaning when they're not proximate to each other, the chances of successful reproduction are very low. And so when we find these guys across acres of bottom and we come out with 39, it's telling that the reproductive process is hindered there. And we found no small clams, only those large adults.
Great, thank you for that. There are 2 more questions. If you want to answer them directly in the chat, in the interest of time, we'll move forward. Our next presentation is a student presentation, so judges be ready. Restored Coastal Habitat in the Indian River Lagoon Can Reel in Juvenile Sportfish by Jennifer Locke.
Perfect. Do you see my first slide? Okay, sorry about that. Thanks everybody. There's been some really great talks so far. And I'm sure this audience is no stranger to the fact that estuarine habitats are really important. But as it pertains to fish, they're functionally connected to each other, as well as offshore habitat via fish movement, which links inshore nursery habitat to offshore adult habitat, which can support things related to like spawning and foraging, as shown in this figure by Gilby and colleagues in 2018. This crowd is also well aware of the fact that coastal habitats are in decline worldwide, and this can lead to declines in fish populations, including sportfish. Habitat restoration can act as a potential solution to address habitat-related declines in fish, and previous studies that have looked at the fish response to habitat restoration have found that they can improve abundance and production. However, fewer studies look at these higher trophic level species like predators such as sportfish. And as I define sportfish, they are these higher trophic level fishes that are targeted by anglers. Therefore, they have an important ecological role due to their position in the food web, but also they're economically significant, particularly in Florida, where they support a $6.6 billion economic impact through recreational fishing. And they're particularly important both historically and today in the Indian River Lagoon with this favorite pastime of fishing. So the objective of this study is to assess the impact of habitat restoration on juvenile sportfish populations and communities. So I first want to go over the restoration methods utilized here, which are the goal of converting a dead oyster reef to a live oyster reef over time by raking down these dead piles of dead oyster shell to the appropriate tidal elevation. And then we secure oyster mat, which at the time consisted of a plastic mat with oysters attached to them and anchored to the site with concrete donuts. and they do recruit live oysters over time. And David actually described these reefs beautifully, so he just presented some data on that. I did also want to discuss some of our living shoreline results, but for the sake of brevity, I'm just going to focus on oyster reefs. This study takes place in the northern Indian River Lagoon system in Mosquito Lagoon, where we have 4 reefs that were restored in 2017, 4 reefs in 2018, and these are paired with 4 dead reefs that served as the negative control and 4 live reefs that serve as a positive control. And they're shown in the blue circles on this inset map, just to give you an idea of the distribution of the sites across the area. As it pertains to fish, we sample fish using a 21-meter haul bag seine net before restoration, every 2 weeks after restoration, and then every 3 months to 6 months for up to 3 years thereafter, in a before-after control impact experimental design. All the sport fish that are collected are enumerated and identified, and then they're also compared to other fishes that are caught at that time. We also look at the biotic data of the reef, being the oyster count and the reef height, as well as abiotic data at the time of sampling, including temperature, dissolved oxygen, salinity, water clarity, and the tide and weather conditions. The way that we look at the influence of these predictors on the abundance and diversity of sport fish is utilizing generalized linear models in the MASS, LME4, and vegan packages in R. So getting right into the initial abundance results, for these figures, I have the 2017 reefs in the upper panel and the 2018 reefs in the lower panel. The red color represents dead reefs, the blue are live reefs, and gray are restored. On the left-hand side of the figure, we're looking at mean sport fish abundance, so that's mean across the number of replicates per site type over time. And what we see is that sport fish abundance is pretty variable over time, which is unsurprising, but that restored reefs are performing pretty well in attracting sport fish, and they are looking pretty similar to live reefs. When we pool these data on the right, over time, we're actually seeing that restored reefs accumulate more sport fish than the controls do, which was a little unexpected. We predicted that they would be intermediate to these 2 controls. So when we look at these data in relation to the oyster metrics, I have the same setup for our figure here with years and colors, but the left-hand side of the panel is showing live oyster count on the x-axis, and the right-hand panel is showing the reef height in millimeters. And the same thing with sport fish abundance on the y-axis. What we see is that there is a negative relationship between sport fish abundance and these oyster reef metrics at dead reefs, which is unsurprising, as I just showed you on the last slide. We really don't catch a lot of fish at dead reefs, and there's not a lot of live oysters at dead reefs. And then there's a weak positive correlation between these metrics and sport fish abundance at live and restored reefs, and they do look similar to each other. Giving you just a taste of the diversity, I do want to mention that we have found 9 species of sport fish associated with the oyster reefs, but the 3 most common species are mangrove snapper, lane snapper, and snook, shown here. And for this figure, it's the same setup again with years and colors. The left-hand panel is showing mean species richness over time. And similar to abundance, it's pretty variable over time. And the restored reefs are also doing a good job of attracting species diversity and tracking similar to live reefs. The right-hand panel is pooling this data, but utilizing the mean effective number of species, which is called JOST richness, which basically takes the exponential of Shannon's richness or Shannon's diversity and puts it back into a real number. And a similar trend is taking place here, where the restored reefs do have an overall higher number of effective species than the controls do. So when we look at what's influencing this using the GLMs, we predicted that the reef metrics like oyster count and height and prey abundance would be the most influential for what's driving sport fish abundance. But since we saw such high numbers at restored reefs, we also examined the data for some other influential factors. And the most plausible model for what's driving sport fish abundance is oyster count, reef height, inlet distance, invert abundance, and then inlet distance and dissolved oxygen. And I just want to show this figure here because I thought inlet distance was an interesting factor. So on this figure, I have distance to the inlet on the x-axis, again, mean sport fish abundance on the y. And what we're seeing is that there is a higher sport fish abundance with declining distance to the inlet. And just to orient you that more to this system, the oval areas are approximate study area, and the arrow is pointing to Ponce Inlet. So perhaps there's some influence of the inlet driving larval flow into the area, with the closer proximity reefs to the inlet, having a stronger current that can drive this larval flow, or even just adult fishes that are coming in to spawn and generate the production of these juvenile fishes. So it's an interesting thing worth exploring. For the 2018 reefs, we actually saw the same most plausible model looking at oyster count and height, prey abundance, inlet distance, and DO. And since restored reefs are performing so well compared to controls, perhaps there's some sort of intermediate complexity to the habitat that they really like, but that's another area that's worth exploring. So to summarize, sportfish abundance is variable over time, which is expected, but they are collectively higher at restored oyster reefs, which is great news and exceeded our expectations. And what we're seeing is that the habitat features indeed are important in recruiting sportfish and probably important in recruiting their prey that support sportfish. And also that site location as it pertains to the inlet may be an important factor to determine restoration success for sportfish. So these data can help us better understand how restoration can be utilized as a management tool to enhance sportfish populations in the future, so we can have healthier habitats and happy anglers like you see here. So with that, I want to thank my committee, my lab at UCF, and my department, as well as my funding partners.
Thank you for that presentation. I don't see any questions in the chat right now. If anybody has any, please feel free to type them in. We still have a little bit of time. And if not, we'll just move ahead with the next presentation. This is Chelsea Nietzsche, Effect of Biodegradable Material on Oyster Reef Restoration and Biogeochemistry. If you're speaking, we can't hear you.
Sorry about that. Can you hear me now?
Yes,
we can.
Okay. Hello, everybody. Thank you for the introduction. I'm sorry about that technical difficulty. There have been so many great presentations today. My name is Chelsea Nietzsche, and I would love to acknowledge my co-authors today, Joshua Sachs, Dr. Linda Walters, Paul Sachs, and Dr. Lisa Chambers. Now, to begin, I would like to familiarize you all with the two restoration materials focused on my study. Traditionally, oyster reef restoration efforts in the IRL have utilized an aquaculture mesh material called Vexar, as shown here on the left. And these single-layered mats are composed of durable, extruded polyethylene plastic. However, as growing concern has accompanied the introductions of plastics into the environment, especially in the form of microplastics, the production of alternative materials have been on the rise. A company based out of the Netherlands called Biodegradable Ecosystem Engineering, otherwise known as BEES, as you see here on the right, has produced a mesh sheet called BEES Elements to aid restoration efforts without the use of plastics. The BEES Elements material is composed of biopolymer solanol. It's a biodegradable plastic resin made from potato starch, and cellulose produced as a waste from the lumber industry. Each mesh sheet can be layered and clicked together to create multi-layered beads mat structures, as you see here. For this study, we wanted to pose a few questions to determine if BEES material is equivalent to the Vexar mesh in terms of supporting oyster recruitment, and to determine if and how rapidly BEES degrades over time, and if that is a result of microbial activity. Moving along, all of our sites are located within Mosquito Lagoon and can be divided into northern and southern reefs. We have a total of 8 oyster reefs, 4 residing in the north region, as you see here on the left, and 4 residing in the southern region. In each region, we have 1 oyster reef per treatment. Those treatments include a reef restored utilizing Vexar plastic, a reef restored using the BEES Elements material, 1 live natural reef as a positive control, and 1 dead reef as a negative control. So the first thing that we wanted to make sure of is that the BEES material was actually recruiting at the same rate of the Vexar plastic. And so we did a small-scale field study and measured oyster recruitment rates. To quantify these oyster recruitment rates, oyster count measurements were conducted in 10 randomly placed quarter-meter square PVC quadrats per each reef at each sampling point, at each sampling period. And oyster density was calculated by dividing the number of live oysters by the quarter-meter square area that they were counted in. Data were analyzed in a two-way ANOVA, and oyster density served as the response variable with region and treatment as interactions. From the results, it is evident that within each region, dead reefs are significantly different than all other reef types, and the two restored reefs, that is the bees and the vexar, paired with the live reefs, are not significantly different from each other, especially within each region. The key finding of this study indicates that bees elements restoration material actually does promote oyster recruitment equivalently to traditional plastic vexar mats. Now, bees is a biodegradable material made from the potato starch and cellulose, and its structure is very similar to the organic compounds you see here on the left. But since organic materials do contain large amounts of carbon, we wanted to quantify the amount of total carbon and nitrogen within the bees. As you can see, carbon accounts for 50% of the material, and this carbon could potentially serve as a tasty snack for microbial communities and facilitate microbial degradation of the material. We also noticed that it does even contain measurable amounts of nitrogen. Additionally, it is important to keep in mind that bees is an organic substrate and it is made to decompose over time, which prompted us to look into determining exactly how does the mass loss of bees material over time compare to the vexar plastic material when subjected to the mosquito lagoon field conditions. To do this, we placed 40 litter bags containing uniformly fragmented bees material of a known weight on 2 live natural oyster reefs located at southern and northern ends of Mosquito Lagoon. Each bee's litter bag was paired with a vexar plastic control litter bag as well. All bags were deployed at the reef sediment surface, and after 6 and 12 months had elapsed, the litter bags were collected and the remaining mass of both materials was measured. Mass loss was quantified and the average decay rate was also calculated. Data were analyzed in a 3-way ANOVA. Decay rate as milligrams per day served as the response variable with material, month, and region as interactions. As shown in the figure, bees material lost about 10% of its mass after 12 months, whereas the vexar plastic did not lose any mass. Also, there was not a significant difference among the decay rates between north and south locations. The decay rate of the bees material decreases over time, with the rate after 12 months being significantly lower than the rate at 6 months. This indicates a rapid short-term initial decay rate that has decreased over time. To elaborate on the top right photo that you see here, after the bees material had been out in the field for 6 to 12 months, discoloration of the previously white material was very evident, suggesting that microbial communities were actively colonizing the material. After analyzing how bees responded under field conditions, we were curious to determine if bees could serve as a substrate for microbial respiration in ideal conditions. To test this, 20 replicate microcosms, all of them were filled with IRL site sediment, and 4 treatments listed here were constructed and CO2 production was measured over the course of 10 days. The production rate of carbon dioxide was quantified. Now, CO2 rates for both site water and DI did not differ, as shown here in this red box. However, all treatments containing bees were statistically significant compared to all treatments without bees. In fact, in just 10 days, the bottles containing bees material produced 47 times more carbon dioxide than the bottles without bees. This increase in CO2 suggests that the microbial community has actually utilized the bees material as a substrate for microbial respiration. And you may be wondering, well, where did these microbes even come from? Well, these results suggest that the microbial population that is degrading the bees material has come from the IRL sediment. Again, this is a short-term response. These bottles did only run for 10 days, so further investigation of this interaction is needed to determine more long-term results. And so in terms of restoration efforts, biodegradable restoration materials, specifically bees elements, has shown to be an equally viable method when compared to the traditional plastic vexar restoration material. But it is important to keep in mind that this is a biodegradable material, and our data does show that microbes can use it as a substrate, but that the rate that it degrades long-term is still to be determined. And it is important to study this material long-term to determine the longevity of the bees within the IRL conditions, and to further investigate what happens to the oyster reefs when it does start to significantly lose mass. Lastly, really quickly, we are in the process of conducting a leaching study to determine if and how much nitrate, ammonium, and phosphate that the bees material could potentially leach out into the environment in a short-term 5-week study. So I just want to say thank you all for your attention, and I'm very happy to take questions now.
Thank you for that. You do have a question in the chat. Chelsea, any plans to look at what microbes are involved? Thanks for the interesting presentation.
Thank you. I think that would be a great thing to look at in the future. As for my current part in this, there's no plans right now, but hopefully in the future, if we get some funding, that would be very interesting to look into. Anything else?
Okay. No more questions coming up in the chat. So thank you for that wonderful presentation.
Thank you.
Now we'll move on to our last one of the session. This is a student presentation by Abigail Jering, Decreasing Nutrient Concentrations in the Indian River Lagoon with One Isolated Bottle Cap at a Time.
Hello, thank you for the introduction. I'm Abby Gehring, and I'm a graduate student at Florida Tech working with my advisor, Dr. Austin Fox. And I'm really excited to talk to you guys today about a treatment system used to lower nutrient concentrations in the lagoon. So while working on a National Estuary Program funded project to treat water using dissolved oxygen, we were approached and asked if we can make it better, and we said, challenge accepted. And so, as we've already seen from today, the Indian River Lagoon, it does have a lot of problems with eutrophication. So to keep it short, excess nutrients are bad. And so how do we mitigate these excess nutrients? There are existing systems like wastewater treatment facilities, recirculating aquaculture systems, and large marine aquarium that utilize natural biological processes known as denitrification, nitrification, and anammox. So essentially, with nitrification, it's an aerobic process in which ammonium is converted to nitrite and then oxidized to nitrate. And from here, we have our denitrifying bacteria come into play that work to reduce nitrate to nitrite and subsequently to N2 gas. Now there is another process, a more energy efficient process that directly oxidizes ammonium to N2 gas using nitrite as the electron acceptor rather than oxygen under anoxic conditions, and this is known as anammox. And so these systems utilize media to cultivate bacteria on. And so we've tested a lot of different media types and found that with our application, these plastic bioballs over here on the left actually worked very well. But rather than utilizing these, why not try plastic bottle caps? And so why bottle caps? These are made from high density polyethylene and polypropylene material compared to commercially available products that are also made from high density polyethylene. Now these are food grade plastics, and so they're BPA free and they are readily available globally. And so we are thinking of applications even beyond the lagoon, and bottle caps are essentially available everywhere. So the system could be applied to remote and underserved regions around the world. With commercially available products, you do have to increase your plastic production if you want this media type. The specific surface area of bottle caps is up to 400 meters squared per cubic meter compared to the specific surface area of these engineered materials that is up to 500 meters squared per cubic meter. So we're not that far off. And so in order to do some rapid experiments, we used sequencing batch reactors and cultivated bacteria in fixed bed bioreactors for various amounts of time. So essentially, all I'm trying to say is that we had pumped water in, we let it incubate with the media for a specific amount of time, and then we pumped this water out. And so we then switched to a flow-through bioreactor, and we were able to get better results because there's always a supply of nitrogen in it that enabled us to see continued decreases. So rather than just pumping water in and out like the batch reactor, we want this water to just keep flowing, just keep flowing, just keep flowing. And so with this system, we are able to take data samples from both the influent and effluent material. But not only that, is we're able to take samples from these kind of intermediate reactor cells. And this gives us a better understanding of what processes are happening all throughout. And so applying this in our lab, this is our laboratory flow-through bioreactor system, and it's following the same concept of just keep flowing, just keep flowing, just keep flowing. And so one important aspect of these experiments is that we're able to control and change dissolved oxygen concentrations in each of these cells to increase the efficacy of these denitrification processes. And so our results this far are showing that in most cases, we are seeing a greater than 90% decrease in ammonium concentrations in as little as 8 hours. And so we are in fact seeing the removal of nitrogen, but there's also a lot more complex chemical processes that are happening in this time. So taking a look at nitrogen speciation, I just want to point out that the different sizes of these graphs are simply representing that we're starting with overall high concentrations and ending with lower concentrations, but we're still showing how these different speciations have changed. And so for this specific graph, we're looking at different nitrogen speciations in typical lagoon water. And so you can see that we are removing virtually all of the ammonium, and what is left is pretty much all organic. And so there are a lot of interesting and complex chemical processes that occur in this kind of middle phase, but we don't have time to talk about that today. But big picture is we are able to remove virtually all of the ammonium. And so some of the applications of this can be targeted to critical areas. For instance, we have been testing this system with stormwater runoff, lagoon water, and also dredge slurry. And so if we were to take a look at something like dredge slurry, the speciation of nitrogen is a little different, and we're seeing greater than 80% of ammonium, less than 20% of organic nitrogen, and just a little bit of nitrates coming into the system. And again, we have these complex processes going on. But essentially, the big picture is that we are again removing virtually all of the ammonium, and we are left with organic nitrogen. And so overall, kind of the key takeaway from this is that we are starting with these higher concentrations, but at the end, we are able to reduce or decrease these concentrations of total dissolved nitrogen by greater than 70%. And so one important thing to consider when you're doing these laboratory experiments is it scalable? And to answer this question, yes, it is. This is our fully functioning 5,000 gallon treatment system. And so I'm sure the question that's already in the chat somewhere is this lady absolutely nuts? What about microplastics? And so I assure you that we do, in fact, care about the environment, and we are extremely aware of the microplastics issues. And so the main variables that contribute to these secondary plastics is ultraviolet radiation and physical forces like waves. And so you can see in our system, or actually you can't really see, is that it's filled with bottle caps, but all of these caps are completely submerged below the surface, and they are also completely entirely shaded and protected from ultraviolet radiation. And something else that I really want to emphasize is that it is 100% contained. So these caps are in a contained treatment system. We are not just putting them in bags out in the lagoon. We are monitoring them closely, and we're also protecting them from any possible physical forces. And so we're confident that we're not creating any of these second microplastics, but to enhance this confidence, we have been following NOAA's standard methods for microplastic counts and analyzing influent and effluent samples. And so this is just an example of a batch of samples that I've analyzed. We look at this weekly, and so far we are not seeing any significant difference in the microplastics count when comparing influent concentrations versus effluent or water. And so if anything, we're actually seeing less microplastics leaving the system so far, which has been really interesting. And so now I'll shift my question from is it scalable to is it successfully scalable? And in short, yes, it is. And so we're once again taking a look at ammonium concentrations and how much they've decreased. And so you can see that once again, we are removing about greater than 70% of ammonium in most cases. And so we are seeing similar trends in our nitrogen decreases, and there are several ways to validate that we are seeing denitrification versus just creating biomass. And one of these ways is through looking at our N to P ratios. And so as you can see from this graph, we are preferentially removing nitrogen over phosphorus, and this helps to confirm that we are seeing these denitrification or anammox processes. And so one of the really amazing things about this project is that we have just been absolutely blown away by the public support and the public interest with this. And so this is a map just showing some of the local schools that I've been involved with and in contact with because of this project. And through this, I've been able to educate the students about lagoon science, getting them engaged with our research efforts, and also raising awareness about plastics in the environment so people can start to keep track of their own plastic consumption while also being educated about the problems that we're seeing in the environment with plastic. And so these schools have just really taken off with this. They've gone above and beyond, making just amazing donation bins, flyers, posters, everything you can think of. We've also worked with Promise and Brevard, and they've set up a lot of collection bins around their community. up to Bethune Beach with Beach Relief. They're really focused on keeping our beaches clean. We've collaborated with local businesses like the Sun Shop Cafe, as well as the Tiny Turtle and Coco. And Brevard Zoo has been a huge help with the donation process. And again, we are just blown away by the public support. And it's been a great educational opportunity to kind of get these younger students involved. I've been working with schools in Miami, New Jersey. We've gotten people reaching out from Canada, all over the United States. And so the potential for outreach has just been phenomenal. And we're really excited to move forward with this project. And with that, I would just really like to thank our lab tech, Stacy Fox, for helping with all of this nutrient analyses. I also owe an immense thank you to my advisor, Dr. Austin Fox, for always sharing his passion and his care for helping the lagoon, and for his patience and guidance along this research endeavor. Like I said in the beginning, this project was kind of born out of a project funded by the NEP, and so a huge thank you to them, as well as Dan over at Lapin Services for his constant support, and the City of Cocoa Beach for allowing us to have our pilot study. Really, I would just like to say thank you to everyone, each individual, school, organization. We're just blown away by the support, and it's really appreciated. We're super excited to see where this goes in the future. And so with that, I'll leave it to you guys. If there's any questions, comments, concerns, feel free to reach out to me at my email. I can catch up with you in the chat later on, but thank you for listening.
You have 7 or 8 questions in the chat and a bunch of congratulations. So if you want to go back through there and answer them there, since we're out of time for this session, but great presentation. And with that, we are moving into the 3-minute slides, and I will turn it over to Dennis.
Okay, so just remember that we don't have any questions during the 3-minute sessions, but you're more than welcome to ask the questions in the chat, and then the speakers are more than welcome to reply to them. And all the speakers know they have 3 minutes, and we have control, so we can just turn you off. We don't have the big hook this year. We don't need it. We'll do it all virtually if need be. But I'm sure everybody will be right on time. The time starts, as they all know, when I say their name. So first up, talking about the cyanobacteria blooms is Connor. You're up, Connor.
Thank you. So my lab group monitored the blue-green algae bloom of late last year in the northern IRL. To achieve this, we measured densities of blue-green algae containing the accessory pigment phycocyanin via flow cytometry. We had 3 main objectives in our study, the first of which was to monitor temporal progression of the bloom in the IRL and BRL. The second was to monitor the spatial expansion of the bloom, and the third was to monitor the bloom die-off. Next. And so what we found is that in the northern BRL near Merritt Island, cell densities of blue-green algae with phycocyanin were relatively low in July and increased gradually through September until leveling off at around 3 to 5 million cells per milliliter, up all the way through November, as displayed in the graph to the left, which shows a really nice growth curve. In contrast, the southern BRL near Pineda Causeway showed little growth and remained at relatively low densities. So we don't believe the bloom reaches far south as Pineda Causeway in the Banana River Lagoon. Unfortunately, we were unable to collect additional data past mid-November due to an outbreak of COVID in our lab. Next. Moving to the IRL, back one. Moving to the IRL, if you look at the graph on the top left, we saw moderate densities of the blue-green algae with phycocyanin and MIMS at around 2 million cells per milliliter and very little change from September to November, suggesting little growth. If you look at the graph on the top right, we saw relatively high densities of blue-green algae with phycocyanin in the IRL around North Merritt Island at around 3 to 5 million cells per milliliter and a slow decline from September to November. So it looks like we missed the growth of the bloom in the northern IRL, but we monitored the peak. If you look at the graph on the bottom left, in the IRL near Pineda Causeway, we saw relatively low densities of blue-green algae with phycocyanin in late September, and then a rapid increase over a 2-week period until reaching around 3 to 5 million cells per milliliter until the beginning of November, and then perhaps dying off towards mid-November. And lastly, if you look at the graph on the bottom right in the IRL near the US 192 Causeway, we saw relatively low blue-green algae with phycocyanin densities from late September until mid-November, suggesting the bloom did not reach as far south as US 192 in the IRL. Next. And so anecdotally, what that looked like in the IRL was a moderate turbidity in MIMS, high turbidity in North and South Merritt Island, and low turbidity around the US 192 causeway. Next. And so the big takeaways from this are that the southern IRL near Pineda Causeway and the northern BRL near Kelly Park and Merritt Island both showed rapid bloom growth and leveling off around 3 to 5 million cells per milliliter, and also that the bloom had greater spatial expansion in the IRL, reaching as far south as Pineda Causeway. And so we were able to meet our first two objectives, looking at the spatial and temporal progression of the bloom, but we fell short of our third objective due to an outbreak of COVID in our lab. But anecdotally, we saw the beginnings of fish kills and anoxic conditions in the IRL mid-December. Thank you.
Well, thank you, Connor. Next up, we're going to hear about phytoplankton blooms in silica, and that will be Amanda.
So, as many of us have become aware, a bloom event which began over the summer took place in the Northern IRL. I took advantage of this event for my senior field project at FIT, with the primary goal to investigate the role of silica and how the availability and ratio of silica to other nutrients influence the composition of the phytoplankton community. These trends were examined at two sites, one in the Indian River Lagoon and one in the Banana River Lagoon on the northern side of the Pineda Causeway in Melbourne. I chose to focus on silica because diatoms compose a dominant group of phytoplankton in estuarine and coastal environments. Unlike many other types of phytoplankton, including many harmful taxa associated with blooms in the IRL, diatoms require silica as an essential nutrient. In order to observe these shifts, sampling was repeated 4 times at each site from September through October 2020. The satellite image I've included on this slide was of the bloom shortly before I began sampling in September. Samples were composed of water quality data, plankton tows, syringe-filled water samples for nutrient analysis, and whole water samples for flow cytometry. Next. These graphs illustrate average cytometer cell counts for small non-diatom phytoplankton versus silica concentrations on the left, and on the right, small non-diatom phytoplankton cell counts versus the silica to nitrogen ratio. IRL sites are symbolized in red and BRL sites in blue. Since the bloom originated in the northern IRL, we can see that the cell counts were consistently higher at the IRL site. In the graph on the left is a regression for which we can see that as silica concentrations increased at each site, cytometer cell counts also increased. And the graph on the right is another regression for which we can see that as the ratio of silica to nitrogen increases, the average cell counts also increase. At this time, microscopic analysis for CHO samples has yet to be completed. Therefore, the exact proportion of diatoms and other larger groups has yet to be determined. However, these increasing cytometer cell counts when silica concentrations are high and silica to nitrogen ratios are high could correspond with a decrease in silica utilization by diatoms, as other non-siliceous algal species experience increased growth rates with eutrophication. Next. An increase in cells containing the fluorophore phycocyanin was also observed, which is indicative of phototrophic cyanobacteria. Each of these graphs indicate the total cytometer cell count in red and the phycocyanin-containing cell count in blue for each site over time. It is clear from these figures that the cyanobacteria were the dominating cause of this bloom. The occurrence of cyanobacterial blooms in subtropical systems is consistent with the long residence times and accelerating eutrophication of the northern IRL. Thank you.
Thank you, Amanda. That was well timed. Well done. Next up, we have Tim, who will talk about evolution.
Am I good?
Cyanobacteria bloom.
Hello, Tim Moore, Harbor Branch student of life. One of the most conspicuous features of the IRL in 2020 was a cyanobacteria bloom that lasted from July through December. This was largely confined to the north and the 3 subbasins of the IRL, the Banana River, and the Mosquito Lagoon. Although not positively identified by us, we believe this was a small single-celled cyanobacterium with gas vacuoles and was the dominant organism in this bloom. Using buoy, satellite, and environmental data, the timing and location of bloom dynamic aspects were reconstructed. Next. The three plots on the left show environmental conditions during the early stages of the bloom and span from May to September. From top to bottom, the plots show daily precipitation, river flow, and salinity. All three show that towards the end of May, a rainy period started and continued through the summer. River flow increased an order of magnitude. Salinity steadily declined in all three subbasins. Chlorophyll in the upper right shows the western IRL was the first basin to show an increase in chlorophyll, which is the green line, with a peak in mid-June and mid-July. It's unclear if these two peaks were from the same organisms though. The Banana River showed a peak that followed in mid-August, and the Mosquito Lagoon showed a more gradual rise into September. Next. This sequence of true color images from Sentinel-2 spans from mid-August to the end of October. In these images, bloom areas had a distinctive greenish appearance that was also visible to the naked eye. The bloom appeared in the Banana River in mid-August by the barge canal connecting to the western IRL. This bloom then spread to the north and south in the Banana River and was fully developed in all three subbasins by early September. So it appears this cyanobacteria bloom started in the IRL and then spread into the other regions. The timing of the bloom in the Mosquito Lagoon is less clear. Next. These plots show the time series at the end of the bloom from October to the end of December. The left plots show chlorophyll and oxygen saturation. The distinguishing feature from these plots are the timing and sequencing of the chlorophyll decline in the western IRL and the Banana River, with the Banana River again following the IRL. These declines were linked to oxygen depletion in each basin, which reached near zero first in the IRL and a few weeks later in the Banana River. Next. Satellite imagery from December time period captures the collapse in the Banana River. In early December, the bloom was still strong while it had already collapsed in the IRL. Dark discolorations in the water began to appear at the eastern end of the barge canal near the center of the second image. A few days later, a large coherent segment of the Banana River turned brown, and cell counts revealed a high abundance of heterotrophic flagellate. This sequence aligns with the chlorophyll and oxygen depletion observed from the buoy data. By January, the bloom is over. This work was conducted as part of Harbor Branch's Florida Center for Coastal and Human Health. Thank you.
Thank you, Tim. Several people have offered to see if you will advance their slides for them. They think they do that in a very nice way. So next up, Rachel is going to talk about primary producer shifts during COVID. Rachel.
All right, thanks, Dennis. So today I'm going to talk about the primary producer shifts that we observed in the northern Indian River Lagoon and the Banana River during COVID. And the previous talks have really been a good setup for that because those are kind of the after. And so I want to get to a little bit of the before the big bloom. And first, I want to acknowledge my co-authors, Diana Bellotti and Brian LaPointe. So the motivation for this study was that we went out sampling last year, and there were Chlorpa prolifera blooms just as far as you could see throughout the northern Indian River Lagoon. And as Dennis mentioned earlier this morning, the water was clear, and we hadn't really seen anything like this in this area, and so we wanted to know more about what this meant ecologically. And this is because if you see the little inset picture I have here, Chlorpa prolifera is a rhizophytic macroalgae. It looks a lot like seagrass, and we wanted to know what these giant blooms meant to the lagoon. So we came up with this great plan to study this question. We had 5 sites that we were going to sample quarterly, look at the benthic cover, the chlorophyll density, and the faunal composition and density. However, what happened is that we sampled in March, and we had to stop sampling because of COVID. And then we didn't get to sample again until November 2020. Next slide. So there is a big difference between March and November. So basically the water clarity and benthic cover were completely opposite. So in March of 2020, we had Secchi depths greater than 1 meter. And this was at sites that were about 1 meter depth. So top to bottom visibility and complete 50% to complete cover with Chlorpa. And then compared to November, we had Secchi depths as low as 0.2 meters. So basically, if you put the Secchi disc in the water, it disappeared. and we found no evidence of SAV at these sites. So as you might imagine, the community composition that we observed was quite different between these two time points. So we sampled the benthic macroinvertebrates and resident fishes with a modified Newston net. And what we found was much higher density of individuals in the spring, 1,800 versus 250 in the fall, or 240. And we also had a lot higher diversity, so 13 taxa in the spring versus 8 in the fall. And while there was some overlap with the eelgrass, isopod, and gobies, on the whole, we saw a lot completely different community composition between these two time points. And so what we can preliminarily conclude from this is that these different primary producer types and densities will have ecological effects. Next slide. But we don't want to draw any conclusions from this from only two time points. And there are other factors to consider, such as seasonality. So moving forward, we'll sample water quality and benthic cover monthly in 2021 and sample fauna quarterly. And thank you to all the people who helped with this.
And thank you, Rachel. We're going to switch into restoration now. We're going to hear from Katie on resident fishes and as indicators of oyster reef restoration success. Katie.
Thank you. As an ecosystem engineer and a foundation species, oysters support the base of a much larger food web when they form a reef. So naturally, oyster reef degradation can have negative cascading effects on species that are reliant on the reef, such as reef residents, which are small fishes that heavily depend on the reef for food, shelter, and nesting opportunities. And oyster degradation has resulted in increased restoration efforts, and a major benefit of those restoration efforts is the enhancement of essential fish habitat. And since the residents also fill an intermediary role in the broader reef food web, they can be good indicators of successful restoration. To further investigate this, we've compared the response of reef resident fishes to restored oyster habitat with standard metrics of oyster restoration success. So sampling was conducted over a 2-year period beginning in the summer of 2017 through to the summer of 2019. And on the map there, we can look at the sites, the locations of all the reefs that were sampled, which includes 7 restored reefs, and then 8 controls, 4 of which were dead, being the negative controls, and 4 were living reefs for the positive controls. And next. So across the 2 years of sampling, 1,033 individuals representing 8 species of reef resident were collected. And species compositions were similar among all reef types, with 7 of the 8 species occurring at each reef type. Live reefs consistently had the highest abundance of fish, followed by dead and then restored reefs. So mean live oyster count and mean reef thickness were the metrics used to characterize structural differences among reef types. And we can see by the figures on the right that immediately following restoration, the reef thickness and live oyster abundance at restored reefs, shown by the green line going up, was pretty similar to the measurements for dead reefs early on. And then about a 1.5 years following restoration, reef thickness and live oyster abundance at restored sites have steadily increased, showing restored reefs basically transitioning from a dead reef to a live reef. So using reef thickness and live oyster abundance as predictors of reef residents' response is more effective than using reef type alone would be. So with these, next. And then with these two figures, we can see reef resident abundance and species richness, both of which increase as oyster reef thickness increases. And then these results emphasize the importance of interactions between reef-associated species and the structural complexity of the reef itself. And while the thickness appears to be a major driver of resident fish abundance, more time and restoration monitoring is needed to see if the fish assemblage at restored reefs shifts to closer resemble the assemblage at live reefs. And this study demonstrated how oyster reef restoration can create essential fish habitat in a relatively short time period. It also suggests that the benefits of oyster reef restoration are transferred from the oysters to the resident community. And this shows the importance of incorporating metrics describing lower trophic level fishes, as they can function as indicators of restoration success.
Thank you, Katie. Please remember to send some good points to Katie, if you can. We're trying to encourage chat, especially for the students. Next up is Anthony. He's going to talk about nitrogen reducing media in regard to septic systems. Anthony.
Good afternoon, y'all. With Brevard County Natural Resources and the Save Our Indian River Lagoon program. We're working on a variety of septic projects where conventional septic systems are a problem. We usually find these septic systems really impacting the lagoon when they're placed in sandy soils, along water bodies, areas of shallow water tables, dense development where their denitrification cannot overcome the loading rate. So what is a conventional septic system? It consists of a septic tank and a drain field, or known as a leach field, and treats about they treat about 30% of the nitrogen within the tank and drain field. The remainder of the nitrogen, typically ammonium, nitrate, nitrite, can impact groundwater and adjacent water bodies. So to address this problem, we're testing alternative passive septic technology as a nitrogen reduction option where sewer isn't currently feasible. Next slide. So currently, the only passive nitrification system allowed by the Florida Department of Health is the in-ground nitrogen reducing biofilter, or INRB for short. So this system modifies the soil below the drain field to promote a natural nitrogen cycling into gas. So the effluent first passes through a nitrifying layer, typically a sandy mix. Then it'll then pass through a denitrifying layer. These layers provide a bacteria-friendly environment without the extra uses of pumps or tanks. So unfortunately, the only denitrifying media currently allowed uses wood chips in an unsaturated environment. So in this situation, wood chips are likely to decompose quickly and potentially contribute to the nitrogen loading issue. So the image at the top shows one of the few systems tested for this new rule. And they're using a layer of wood chips mixed in with sand as the bottom layer of this drain field. So next slide. So the DOH has listened to the concerns with the current INRB rule and entered into a memorandum of understanding with the county to install INRB systems using alternative media that is known to work within Florida. Under this MOU, we must measure the effectiveness of the alternative media, and if effective, it can be approved for statewide use, as long as it meets the rest of the DOH standards for drain fields. So for sampling equipment, we're using 12 pan lysimeters with 6 stage to collect effluent below the nitrifying layer and 6 stage to collect effluent below the denitrifying layer. The lysimeters hold at least 2 liters of effluent and have an overflow to prevent pooling in the pans. So the first 6 alternative INRB systems using Bold and Gold wastewater filtration media are in the ground and functioning. We're looking for another denitrification media to test for the next 6 systems. So hopefully a year from now, we'll have data for you. Back to you, Dennis.
Thank you. We look forward to that next year. Next up is Jenny, alternative oyster restoration materials. Jenny.
Just wanted to give a quick introduction to projects we're working on funded by the National Estuary Project and Save Our Indian River Lagoon Fund, looking at alternative to plastic restoration materials on oyster reefs. We've worked with collaborators at the University of Florida, Whitney Lab, and IFAS, as well as the Brevard Zoo Restore Our Shores team, to investigate 2 different shell density oyster core modules, 2 different gauge gabion, galvanized after welding steel gabions, oyster prisms, and oyster ball at 3 locations along the beaches of the Indian River Lagoon. These were deployed in triplicate and hung underneath docks. And at this point, we'll be monitoring water quality variables such as salinity, water temperature, pH. We have continuous depth measurements and are quantifying percent cover, fouling, deformation, breakage, and corrosion. Next, please. So on the upper right, we have a graph of the continuous depth over the first month. This experiment will run for a little over a year, and we just kicked it off in October of this year. So we've seen some initial recruitment of tube worms, bryozoan, and algae. Most of the fouling was algal detrital material, and the salinity was somewhat variable. Water temperature dropped by a little bit over the course of the first month, and the pH ranged from 7.75 to 8.18. We normalized the degree of fouling by measuring before and after cleaning on each material type, and normalized by the volume of each material. And over the first month, there really was no significant difference between any of the treatment types. Next slide, please. So as we continue this project, we will continue to look at these abiotic factors, structural integrity as by percent cover. We haven't had any significant breakage yet other than the oyster balls. The string that held the shells on was a little fragile, but the rest have held up quite well. And we will be deploying in the spring to look at oyster recruitment and growth, as well as commensal organisms. And then with the outcome of this study, we're hoping to pilot on our restoration sites using a habitat suitability index that we've been developing and will be reviewed by a technical advisory committee. And an example of our new site analysis tool is also there in small print that just shows how much goes into our site selection criteria. So with that, I just want to thank everyone who's been helping with this, all our collaborators and funding agents, and hope to give more information next year.
Oh, that's good, Jenny. You had 5 extra seconds to put to the bank. Thank you. Next up is David, who's going to talk about the lagoon monitoring plan. David.
Thanks, Dennis. The NEP is developing the One Lagoon Monitoring Plan to specifically support the IRL CCMP. The monitoring plan connects to 3 CCMP categories of vital signs, including water quality, habitats, and living resources. Next slide, please. For this presentation, I'm going to focus on existing assets and the resultant proposed One Lagoon Ecosystem Monitoring Network. Specifically, we identified who is collecting data, and for each of those, what the data is comprised of, where the data is being collected, and why the data is being collected. In other words, linking data to the CCMP vital signs and indicators, which should ultimately drive management decisions. Next slide, please. All monitoring programs need to support management of the IRL, must be active, must comply with an established QA/QC program, and must make their data freely accessible. Development of the draft monitoring plan has been accomplished through the input and oversight of a 15-member steering committee. Inventory of existing assets was accomplished by converting inputs from a previous survey and mining existing monitoring program databases to develop a list of monitoring programs. Everyone on the list was sent a questionnaire regarding the who, what, where, when, and whys of their specific program. The questionnaire results were synthesized and presented at a workshop last November. There were approximately 40 attendees who participated in multiple surveys to assist in refining the proposed monitoring plan. Our draft report will outline the proposed ecosystem monitoring network, including a detailed summary of each monitoring program, an extensive series of maps depicting the location of all sample stations, as well as gaps in data and analyses, emerging needs and opportunities, and specific recommendations for action. Next slide, please. The proposed network includes 67 individual monitoring programs spread across 22 different organizations. These include federal, state, county, university, and non-governmental organizations. The proposed monitoring network is much more integrated and complex than the tables on this slide suggest. Many programs in the monitoring network rely on partnerships to accomplish their monitoring activities. Additionally, individual programs may address multiple vital signs across several CCMP categories. 5 of the organizations rely on citizen science to collect data. Long-term outcomes of the monitoring plan will hopefully result in a healthy IRL due to improved design, implementation, adaptation, and accountability for many individual actions. Lastly, I encourage you to email me with questions or comments, especially if you're routinely collecting IRL data and you haven't heard from us yet. Thanks, Dennis.
Thanks, David. And you put some more seconds back into the bank. We like that. Next up, our last speaker of the session is Bob Bernstein with the intriguing question of how many amphipods can dance on a blade of seagrass.
It's a pleasure to be back, even if it's only virtually. Welcome from Gainesville next year, hope. My co-author is Patrick Dowling, a former summer intern at Harbor Branch. This is COVID related in the sense that you could do it on your kitchen table, or you might find a at the bottom of a file cabinet or the back of a closet. In this experiment, we used a 1 liter bowl with one 5 centimeter long blade of turtle grass, the Thalassia testudinum. We added amphipods into it. No additional ones were on the grass blade. We call that saturation. And a note that amphipods are ubiquitous primary consumers of algae, particularly microalgae, and they are tasty. We used 2 species of amphipods, first separately, then added both together, and I'll try to answer so what. Next. This amphipod, Cymedusa, is about 1 centimeter long, and shown are 2 plots versus time. The top line is the number of amphipods added to the bowl. The lower line is the number of amphipods on the seagrass blade. Initially, both numbers climbed. For example, at 10 and 20 minutes, all the added amphipods were on the seagrass blade. But then the number of amphipods on the blade tended to max out at about 20 individuals, no matter how many additional amphipods were added. This density would be a field equivalent of about 300,000 per square meter, but that's 2 to 3 orders of magnitude greater than what we find. Therefore, we might conclude that the space is not a limiting factor for amphipod density. Next. Similarly, this amphipod, Elasmopus rapax, reaches a peak density of about 15 individuals on the blade. It's larger and more robust than Cymedusa compta. The gnathopod, or claw-like appendage, is seen just under the eye. It's described as large and robust, especially in the males. And you might ask, what happens if we put both species in together? So both species were added simultaneously. Eventually reaching their saturation densities. The result, it was a king of the mountain fight. I mean, not just bumping into each other, it was down and dirty. Elasmopus literally kicked the smaller, less robust Cymedusa off. Occasionally, Elasmopus ate one of the Cymedusa. Eventually, there were only Elasmopus on the blades, no Cymedusa. Some moves were incredibly athletic, with Elasmopus lifting a Cymedusa over its head and tossing it. Just like this is what it actually looked like. So we conclude that space is not the limiting factor in the field, but interspecific interactions can alter species composition. And to sum up, as Yogi Berra said, you can observe a lot by just watching. Thanks.
Thank you, Bob. And I want to thank all the speakers. I want to thank all the speakers in the session. You did a fairly good job staying on time. So first, I want to say that we're going to go into poster session mode, and I'll explain that. Some of you, if you want to stay in the main room here, the virtual auditorium, if you will, you can do that. If you want to have more dialogue, you're more than welcome to do that. Otherwise, you will see Wyatt is putting in a series of links to the virtual posters that you can go to. And we're going to have 2 rounds of posters coming up. There's 3 posters rooms for 30 minutes, followed by 3 different poster rooms for 3 other sessions. Now we're going to repeat these tomorrow morning. So remember that you have more than one chance to go to the posters. And we're going to give you, say, 5 minutes from now to get situated. And then we're just going to run the clock for 30 minutes. And then that first poster session will be over. And then we'll go immediately to the next one. And that has another 30 minutes. However, since that is the last event of the day, that will be a soft ending, if you will. So we won't cut those rooms off, after 30 minutes. If you all have a lot of discussion going on and you want to keep going, it's COVID. We got no place else to go. we'll stay here and keep everything open. Okay, so that's where we are on that. And then we're going to reconvene tomorrow morning. So I'll just say that now and say that in about 5 minutes, we'll start each of the 3 poster sessions. So the information is on the screen, and I think it'll also be in the chat.